Good morning, everyone. Welcome to the Chapter 2 of FlowJo High Parameter Data Analysis course. Now, before we begin with the course, there are a few ground rules that we all will follow so that we can have a smooth session. Firstly, please turn off all your other cameras and listen to the speaker attentively. Turn off your cameras as it creates a disturbance for the speakers. All of you have been put on mute, but we encourage you to ask as many questions in the chat window. Also, there will be a link which will be shared in the chat window for marking your attendance. We request you to just put in your names and organization name in that form. The session will be recorded and will be shared only on institutional email IDs, and another form will be shared for the same. Do fill it and we will be sharing it on those. Now, I would like to welcome Ubaldo Barbosa, our National Application Manager, who will be the moderator for the course. Sir, over to you.
Thank you, Ankita. I think we are now into the second chapter of the FlowJo session. Today we have those participants who are going to the next level. So this is the chapter 2 of the high parameter data analysis course, where we are going to get into more details of the FlowJo software. I'm sure in the first chapter we got an overview. Tim took us to very detailed parts of the FlowJo software, where he showed us all the sequence of using the software. Also, he touched upon the compensation modules. And today we're getting into the second chapter, where Amlan Ghosh will be leading us to that journey of the high parameter. So Amlan needs a little bit of introduction. He's done actually. Amlan is a BD person based out of Pune. He's an application scientist based out of Pune. He's trained as a cellular immunologist with nearly 10 years of experience in multicolor flow cytometry. Amlan is very passionate about driving the flow cytometry education, and especially the FlowJo software training as well. He leverages his expertise by supporting researchers in his current role as an application scientist at BD Biosciences. Amlan has done his PhD in immunology at NICD, National Institute of Cholera and Enteric Diseases, India, based out of Kolkata, and has done his postdoctoral fellowship from Loyola Institute of Immunology at San Diego. So with this introduction, over to you, Amlan.
Good morning. Good morning, all of you. Good morning, Peter, Ankita. Thanks for the great, kind introduction. So just confirm that, my camera is on. And
yes, we can see you. I can see you, Amlan.
Okay, great. And you can hear me also?
Yes.
Okay. So not very sure when I'm going to show the software, whether I have to turn off my camera because of the internet issues. I will not take a risk there. So, but for the presentation mode, I'll definitely keep my camera on. So let me pull up my slides. Okay. Can you confirm, please, whether you can see this?
I can see your slides.
Okay, great. Thank you. So good morning, everyone. So today we are going to discuss about high dimensional data analysis employing algorithms for clustering and dimensionality reduction. Excuse me for, I got a cold. So last session, Tim took us through the basics of FlowJo software, introduction to FlowJo, also compensation, and how to install R and plugins in your FlowJo software. So which will enable us to do the dimensionality reduction and all those clustering algorithms. So now today I'm going to go deep into using those plugins for your high parameter data analysis. So I'll just start with the presentation. So what you can see on my slide is basically this is an example that I use. This is like a grant funding cycle. So which basically goes like this. So if you got a grant, I mean you got some money for doing some experiment, then you design an experiment. You'll probably go and collect some samples, and then you'll do a high parameter flow cytometry, and then you will analyze the data. So up to this part, earlier it was okay. After you analyze the data and you got some good results, you go for a publication, and which will, I mean, which where you will try to put up your new hypothesis and which completes the cycle. But now, with the advancement of techniques and tools that you have, you are really pushed more towards, after this data analysis of the high parameter FlowJo, flow cytometry, then you are pushed to go for sorting those populations. If you find some nice, I mean, rare population or a unique phenotype, you will definitely like to sort it, because sorting or kind of enrichment of that population will take you further of all those applications, downstream applications, principal one being multiomics, where people use single cell RNA sequencing and AppSeq to study in depth the transcriptome and protein level expression of different markers on their cells, and then they do the data analysis and then review and interpret, and that goes for a very high level publication. So the difference being just data analysis to publication and doing all these steps again and then going to publication is the impact of journal. Probably you will end up having a high impact journal publication, so which will complete the cycle. So today we are going to focus on the data analysis part, which comes from high parameter flow cytometry. And what you see on the left side is basically the workflow that we try to popularize, and we call it discovery workflow. And the steps I'll take you through one by one. But just to summarize here, so these are the essential, I mean, mechanism that you need to follow once you have some more than 20 color data, let's say, because nowadays people are doing more flow cytometry with, I mean, larger colors and parameters, 20, 25. This is becoming a common practice. So then the traditional manual way of gating doesn't really work when you're really trying to find something new in it, or the traditional gating with the available literature knowledge doesn't really help. So fortunately, we have a lot of this dimensionality reduction algorithms and clustering tools, which help us to dive deep inside those complex population and reveal their phenotypes. So using FlowJo, you can really do a lot with respect to that high dimensional analysis, which you'll see today. So I will first finish my deck, go through it, and then we'll take a short break and maybe some quiz, 2, 3 questions. We'll try to go through them, and then I will go to a live mode where I will show how to use, I mean, few of these plugins with some actual data. Then Tim is going to take it forward with some more advanced analysis on FlowJo. And finally, we may have a small session where if time, if let's see if we get there, if time is there, we can have a live demo of how to install R. And then we will end today's session, and you will have an assignment that you can practice, because all of you have license to FlowJo now, and we have a set of instructions and like 5 FCS files which will give it to you for practice purpose, and you can follow those instructions and let us know how it works. So basically, what we start by doing by having the data is we start gating and doing some cleanup of the data. Cleanup is basically removing all those clogs, debris, dead cells, and also cells which are not of interest. Only the lineage or cell of interest which you are going to work with downstream analysis, you can keep that and remove everything else. So that part is cleanup. There are algorithms which does this for you, maybe Flow Clean, Flow AI, but you can do it for traditional gating also. Then comes concatenation. I'll show that, I mean, in coming slides, what concatenation is. Basically, it is merging of different files into a single file, which goes for the downstream analysis of these 2 steps, dimensionality reduction and clustering. So what it does is, when you do dimensionality reduction algorithm on your data set, you basically reduce the dimension, because it's a high dimensional data, 20 plus parameters are there, but then you try to reduce that dimension and represent it in a lower dimension. So there are algorithms for that. So after you do that, then you make real clusters. So clusters, I mean, when you do the traditional flow cytometry, we do like all those 2-dimensional plots, and then we try to get some clusters. We make some populations, we call it a population A, B. Those thing is pretty similar to clustering what we do here. We really cells which are having similar phenotype, they come close together and form a cluster. So that's a cluster. And while doing all these steps, we also make some keywords. Keywords are basically metadata, which after the clustering analysis is done, we can gate apart different conditions which we created through keywords and compare them, how they change across different experimental conditions. And if you're really interested to go dig deeper, then you have these sort populations. We have tools for that. And also AppSeek, all these applications have fantastic tools. You can, I mean, if you have questions regarding these, I think some part of this sorting, like which requires Hyperfinder, Tim is going to show today. But for AppSeeks and other stuff, if you have queries, you can reach out to us, or you can go to BD Bioscience website. You can have a feel, have a look how it is. There's a lot of stuff on the web. Now, going to the analysis workflow, the left part that you saw just now remains same, but this slide I think Tim showed last session at the very end, just to tantalize you with, or for today's session. Basically, what you see here is, I mean, this is your final population, and these are a bunch of gates, cleanup gates, which have been used to remove clogs, debris, dead cells, and these are all those metadata which have been put in the downsampled event. And this is a concatenated file, which is a merged file. And you can see time point and tissue are two keywords that are used in this particular experiment. So you have tissue 1, 2, 3, 4, and again time point 1, 2, 3, 4. So you merge everything together, and on that concatenated file, you do the dimensionality reduction algorithm. And basically t-SNE is t-distributed stochastic neighbor embedding, which is very popular in the field. And these are the two new derived parameters, t-SNE x and t-SNE y, in which it tries to flatten out your data. And this is the low dimension representation of your high dimension data. That's it. I mean, to keep it simple. And from this, you can really gate apart different conditions like tissue or time point. Like here you can see lymph node, liver, spleen, blood. These are different tissues. So once you have this kind of a dimensionality reduced representation or visualization of your data, you can really see different tissue-wise expression of how your experimental conditions are changing. Like you can see, and you can have a overall idea of any particular area or a population of cell which is getting elevated or depleted, enhanced or depleted. For example, this is a density plot, and you can see this particular cluster is getting enriched in the spleen compared to other tissues. And again, this one is getting enriched in the blood, but not as such in other tissues, maybe a little bit in liver. And these are the basically flow-sum clusters where you just drag and drop all the clusters that you derived out of any clustering algorithm into the layout editor in FlowJo, and it creates actual populations of cells which are having similar phenotypes. They are grouped together, and which have dissimilar phenotypes, they are just pushed apart and form different islands. So that's like a brief summary before we go into the details. Now we go in each step with a lot more details. Okay, so plugins, basically to do all these things, you require plugins. And where do you get it? In FlowJo, you can get it. And plugins are basically like little modular applications that you can download and extend the functionality of FlowJo. What you can see here, I have just put in 4 broad buckets of all the different sorts of plugins that you get, and in different categories, basically pre-processing plugins, dimensionality reduction plugins, clustering, and all those interpretation and deep biology plugins. So pre-processing plugins are some plugins that do something to your data upstream before the analysis. Like downsample, it basically reduces, I mean, make sure that same number of events goes into the analysis. Then cytonorm for longitudinal normalization of your data across different experiments, and flow AI, flow cleaner there, which takes care of like doing some cleaning of your data to remove unwanted events. Now coming to dimensionality reduction algorithms, you have a bunch of them, and this list of plugins is ever expanding. I mean, every month, probably sometimes every week, we are having 1 or 2 new plugins added to this list. So you can I'll show you from where you can get to that updated list and how you can download them. So dimensionality reduction algorithms are basically projection algorithms which I mean compress your data and show it in fewer dimensions. T-SNE, the very popular T-distributed stochastic neighbor embedding. Then you have UMAP. More people are, gravitating towards using UMAP and TriMap. TriMap is a very new one. And then you have also embed sum, which basically projects the flow sum clusters. And in clustering algorithms, you have XShift, flow sum, phenograph, flow means. A lot of them are there, and each one has their pros and cons. Depending on your data and your ask as a researcher, what you want to show, you can use multiple. Which better represents your data, you can go with that. There's no standard rule like you have to use T-SNE always and then this and that. Nothing like that. Now coming to interpretation and dig deeper. So Hyperfinder is like the one which maybe we can show you today if time permits. This is basically if you find some rare population or novel population. These are how you sort them when using the flow cytometer, those populations. So this is basically help you to derive those computationally derived sorting gates. Then violin boxes, there are a lot of different options like heat map, rain cloud for antigen density, violin plots. These are very new features. ISLR can really spin up your data in like 3D view. And Cluster Explorer is really nice plugin, which today I plan to show you some data. I'll spin up in Cluster Explorer after having this clustering algorithm and dimensionality reduction algorithm. These after we do these plugins, perform these plugin actions on your data set, then you can really pull up everything in Cluster Explorer, where it will try to show your different clusters of populations that you derived through XShift in that newly derived low dimensional space, which is there coming from T-SNE. So which is very much like interactive. So Cluster Explorer is you can see the like interaction of XShift clusters in the T-SNE space live. It will come with the principal component line plot and all those heat maps where you can see the different antigen density across all different clusters, and also you can put in your keyword derived the metadata, that is the experimental conditions, how these different clusters are changing across different experimental conditions. So what I've done here is the ones which are in bold italics, so they require the R computational statistical environment for running. So how do you get there? So if you go to the FlowJo ribbon and you can see that there's a workspace tab in it. So if you click there, under which you have populations band, in which you have plugins options. So if you expand this menu, the first line item is FlowJo Exchange. If you click here, it will directly take you to the FlowJo website from where you have all these plugins. And depending on the version you have, like last session, Tim showed through a presentation how you can use the FlowJo, the plugin wizard, which is a basically very new tool that help you to get all those updated versions of all the plugins that are required in your PC, kind of customize it for you. So now going to the details of cleanup gate. Basically what you see here is a clean data. This is my clean data, and this shows the like ancestry or backgating, which tells you that I have started from my singlets where I leave out these doublet cells, and then doing this with forward scatter height versus area, then forward scatter width versus area, again one more round of gating, so that I fine tune on my single cells. Then probably I'll gate my leukocytes, everything, lympho, mono, neutro, everything, and just to get rid of any debris that might be on the edges. Then CD45 gating just to remove non-CD45 cells. And within that, now if in this particular example, I'm really interested to look into the myeloid or lymphoid compartment. So I just cut out my granulocytes by help of HLA-DR versus side scatter gating. So this is a pretty tight gate where I select basically my myeloid or lymphoid compartment and go deep into the analysis of this particular compartment, because this step itself removes 60 to 70% of unwanted events, which you don't really want to put in further down pipeline analysis for all those plugin and stuff like that. So basically, cleanup does not only mean that you remove the doublet, dead cell, or debris all the time, but it also makes sure that you are really focusing on that lineage of interest and removing every unwanted cell, which is a noise. Now downsample, this is used to normalize the number of events which are contributed to that particular concatenated or merged file, which is the next step that we do here. So what it does basically, you can see here, you can select a particular node of events like myeloid or lymphoid here, which is shown, and then you perform this downsample action. This is also located in the plugins menu. So once you do that, it will downsample an event to the particular number that you specify here. Let's say here it's showing 30,000. That means every single FCS files which have this particular node, and whatever number of cells are there, from every file 30,000 of cells will be taken for that next step, and those will be your downsampled population, and it will not select arbitrarily, it will select equidistant event across the acquisition time parameter once it performs that action. So that is how downsampling works. But in the version that we are using now, 10.7 today, probably we can really omit this step. We can do it in the time of doing concatenation itself. I'll show that. So coming to keywords, I'll not explain more on keywords today, because last week Tim showed in a great detail about keywords, how they work, and basically they are sample level metadata which explains different conditions, experimental condition. Like in the couple of slides back, I showed time point, tissue, those two were keywords, and you put like different numeric values to it, like tissue 1, 2, 3, 4, then time point 1, 2, 3, 4, and those will create all those different combinations, and you want to really gate apart. While after doing this clustering algorithm, you can gate apart those conditions and see how the experimental conditions work throughout the different data sets. And they can also be added to FlowJo. The one file that we use today, I'll show you, there are already some keywords have been added. It is like a workspace template file, and the one that you will get, the same one, there you also have already keywords created for you to gate apart. Okay, now coming to concatenation. Concatenation is once you are really ready to go for the dimensionality reduction algorithm and clustering algorithm, so you have to concatenate everything, because what happens is these dimensionality reduction algorithms, they are non-deterministic. That means you cannot do it on individual files and then get some output which are comparable. No, you can't do that. So what you have to do, you have to merge everything together, make a single file, and then do the analysis, the clustering to analysis or whatever on that. And after you do the analysis, from there you can gate apart different conditions, and you can see how these things are changing across your different experimental conditions. Okay, so because these are non-deterministic, you cannot do it on individual files. You have to pull everything together in a single file and then do it. So basically what you see here, different FCS files and different number of events were captured. Once you do the downsampling, the orange bar basically represents the downsampled event, which is a normalization with equally distributed across the time of acquisition. And then all these downsampled events, then you are concatenating and making a single file. That is the concatenated file. And after that, when you're doing this downsampling operation, you can have the new keyword based parameters while concatenating, so that in that concatenated file you can really see those conditions, stimulation conditions, and subject like subject 1, 2, 3, 4, 5, and different stimulation XYZ conditions are there. So later on you can really gate them apart and study. Now, dimensionality reduction, what it does, basically it creates newly derived parameters. So what you see here is like a continent plot. I mean, we call it like continent, and these are different like islands in it. So basically what it does, to put in simple terms, it will allow a visualization of your multidimensional data in fewer dimensions. So you have maybe 20 color data, but depending on the cell types that you have, their relative level of expression of the antigen or particular marker, they are being grouped into these different islands basis on their relative expression. So the cells which have similar expression of markers, they come close together and they form a cluster. Like these are NK cells. So what I've done is like overlaid my actual gated population on this t-SNE plot. So these are monocytes, these are NK, B cells, CD4 and CD8 T cells. So events with a similar multidimensional expression pattern grouped together within the dimensionally reduced space. And these are the two newly derived parameters. This is the t-SNE. I mean, you have seen a lot of options are there. You can do UMAP, TriMap, embed sum. All these are dimensionality reduction algorithms. Some of them are visualization as well. So t-SNE, these are the two new derived parameters, t-SNE X and t-SNE Y, in which it flattens out your data really well, and then it tries to represent like all these different continents, each one representing a particular lineage of cells. So, and it overall tries to maintain a higher order structure. Higher order structure means relation of one cell to the other. So basically here what I mean is, but this is, I mean, in later few slides, I'll explain that there's a problem sometimes happens with t-SNE. t-SNE is not really good in terms of maintaining the global structure. What is a global structure? We'll see that. But you can just, for the time being, you can just, I mean, understand what dimensional reduction algorithm is and how it works. It basically flattens out your data and makes multi-dimensional data represent in a low dimension, where you get all these different populations basis on their overall marker expression. Now these are the different, I mean, representation of the data of the plugins that you have using the same data. So this is t-SNE, this is UMAP, TriMap, EmbedSum. So what you can see is like, so so UMAP is, more people are liking it in doing UMAP basically, and the people who are doing those single cell RNA sequencing and AppSeq data, they're really liking this UMAP because it's fast. It can scale well with lot more parameters in a really short amount of time, and it can handle a lot of events much better than t-SNE. So that's the advantage of UMAP. TriMap is even newer version of like doing a dimensional reduction tool. And what you see here in UMAP, TriMap, these clusters are really far apart from each other. And here the problem that can happen is sometimes same lineage cells can come in three different clusters. So that is the global structures. So sometimes it really t-SNE really fails to keep the preserve the global structure, and same lineage cells can be found broken into all over the plot. So in terms of that, UMAP and TriMap are very good, and TriMap as well. You can, I mean, this is a contour plot, but in like other plots you can able to see there are different trajectories which really tells about the differentiation of between these, this relationship between the differentiation of these different cell types, and also the distance between these different clusters in TriMap and UMAP has a meaning which is really not meaningful in t-SNE. t-SNE, the difference between these islands doesn't mean anything, but in case of UMAP and TriMap, it has a meaning. And now when you consider embed sum is basically when you derive the FlowSum clusters and you want to, because what FlowSum does is maybe it creates hundreds of mini clusters and then merges them to number of meta clusters that user is putting in the UI. And then if you want a grid view of that, of all those derived clusters, then you can have a representation which is called embed sum. So it's basically 2D visualization of FlowSum clustering output. So, okay, now coming to OptSNE. OptSNE is there's a native t-SNE button in FlowJo, the version that you have got. You can open it and try to see. There's a native t-SNE button. So ideally, the standard t-SNE something look like this. I mean, this is an output, but then I mean, with when you put lot more events and lot more parameters in t-SNE, it usually takes a very long time, and then sometimes I mean the output is just a blob of cells. You really cannot differentiate in different clusters. But then we suggested, we tweaked, I mean FlowJo team tweaked within that algorithm to find a better resolution with help of Annabelle Kina. The paper is published in Nature Communication 2019 November. So we have now come up with OptSNE, which is optimized t-SNE algorithm, which is there. I mean, this is now the by default option in FlowJo is the OptSNE algorithm, which deals better with higher number of events and higher number of parameters. Considering the time taken by OptSNE is very less compared to the traditional t-SNE. So now, whenever you are doing t-SNE in FlowJo, most probably you will do the OptSNE, which represents better your data. Now coming to the different dimensional reduction algorithms that you have, OptSNE, UMAP, and TriMap, you can see the global structure is preserved differentially depending upon the method. What I mean is basically if you try to see the same color, I mean this particular color, this is spread in two different positions, and this violet or magenta, this is also spread across the plot in all different locations, which means although these are same lineage cells, they are spread all through the plot. which does not really happen as you use UMAP or even TriMap. Okay, that means it can better preserve the lineages basis on their overall phenotypic marker expression. So this is what I mean by global structure. So I mean, again, there is no standard rule in the field that you have to only use UMAP. You cannot use TSNE, depending on the question that you are answering through your research. That is what is important. So if for some person TSNE works with that data set, then it's fine, and for some other person then UMAP works, then that's also fine. And you can also, there's no rule that you can also use them in tandem. Like you can do the UMAP analysis, then you can select the population, and then you can do a TSNE on that, because TSNE does a good job when it is trying to look at the local structure. That means within an island, you know that you can better resolve different populations, because let's say this is an CD8 cluster, and within that, if you have like activated all those memory phenotypes, it can really resolve well. Okay, so in terms of local structure, TSNE works well, but if you want to keep the global structure more or less preserved, then you should opt for like using either UMAP or TriMap. And as I was telling, in TriMap, you can see all these different trajectories, which has some meaning in terms of differentiation of all those lineages and their relationship. Now coming to when you are really dimensionally reducing your data, what exactly you want to arrive at. So depends on your panel and experiment. So if you are, for example, in this particular example, if you are this person is really interested to look into CD8 cells. So if this is your total like CD45 positive cells, and now if you perform the dimensionality reduction algorithm on this, and this is the output that you're getting, and you can see these are your CD8 T cells which are of interest to you, but you have a lot of unwanted cells in this plot. So the step at which you want to dimensionally reduce your population is very important. You have to go down that hierarchy and arrive at that level on which you want to do that is your cell population of interest, and you want to perform the dimensional reduction algorithm on that. So basically, to arrive at CD8 population and to see different like activated memory phenotypes within that, I would definitely get rid of my granulocytes, then get rid of my monocytes, then B cells, then NK cells, and finally you see this is I'm getting rid of my CD4 cells even. These are my CD8 T cells, which are really, separated well. This is a TSNE, and as I was mentioning, TSNE does a better job in the local structure. So these are the different, islands, and this is a bigger continent. You can see all these different phenotypes. Maybe they represent all those central memory, effector memory, TEMRA, and naive cells or stem cell like phenotypes within the CD8 population. So it does a good job in local structure, but for the global structure, you should opt for other algorithms. So now coming to third parameter color maps, very useful to get an overall idea and orient yourself to the data set that you're working with. Basically, I'll show you that once you have performed the TSNE or any dimensional reduction algorithm, you can have the third parameter color map where you can see one at a time in the graph window expression of a particular marker, like how it goes in through across that concatenated file in that lower dimension space. For example, if I see this data, I can really identify these are my CD19 positive, maybe these are B cells, and yes, they are B cells because they are also IgD positive. So this island represents my B cells. Here are my CD8 T cells, these are my monocytes. Okay, I have CD3, these are my T cell population, and CD123, my plasmacytoid dendritic cells are here. Like this will really help you to orient yourself and identify different like cross populations, how they're distributed across that concatenated file in the dimensionally reduced space. So this is very useful, and I'll show you today while we go live demo with the FlowJo software. Now coming to clustering, these are basically your I'm sorry. These are basically your flow sum clusters. That I mean, there are different options of doing flow sum. Phenograph. Phenograph is a good one. I mean, that's my favorite. But in terms of X shift is also pretty easy. It does not require R even. So basically what it does, when you derive some clusters, actual clusters with similar phenotypic expressions, then you can just drag and drop everything on your layout editor in FlowJo. And then this option is for multi-graph overlay, where you can get a histogram with relative expression of each of the measured parameters in your experiment, and see how they are changing across the different clusters. And for example, CD123 is so the different clusters are color coded differently. So you can identify from the colors which markers are getting expressed, elevated or depleted in all of these clusters, and there's relative expression of all these. So this is really interactive. I mean, when you have a clustering algorithm already performed on your data, it sometimes makes sense to have an overall idea of like, where is my like CD15, where is my granulocytes located, so just to identify in a quick manner. So as I was telling, X shift is there, phenograph and flow sum is there. Flow sum, the problem is flow sum is it's super fast, but you have to tell the software how many number of clusters you want it to find. So that you have to that you have to do the input in the program. But for phenograph, it finds out the number of possible cluster considering the nearest neighbors. And X shift is also does not require R and fast, and it is the method of choice for a lot of people. So today also I will try to show X shift while we do some clustering. And this is the same data. You can see FlowSum does better with big clusters, but for identifying rare populations or small clusters, XShift or Phenograph may be good. So once you are done with the dimensionality reduction and clustering, and you know overall, orientation of how your experiment worked, which all markers got expressed in whichever phenotype. Now you want to gate apart the different conditions. Let's say you have 5 patient data in your experiment, which you initially used a keyword for 1, 2, 3, 4, and 5. These are the patient IDs, PID 1, 2, 3, 4, 5. So each contribute 20% to your concatenated file. So if your concatenated file has 50,000 events, then 10,000 event comes from each. And now if you really want to find a particular population, like this tiny cluster here, what you see is, and you try to see like how this population is getting differentially regulated in all of these patients, 1, 2, 3, and 3, 4, and 5. And you can see really this patient 3 is having a higher proportion of cells here, and maybe this could be linked to some disease phenotype that will be revealed through the other markers in your particular panel. So like the first slide, I showed different tissue, livers, blood, lymph node, like that. Here also you can gate apart through the patients, and you can see in any of the patients whether any particular cell cluster is getting elevated or depleted. So query gate is a very popular option when you have the dimensional reduction done on your data set. You can simply put a query gate and ask what is the phenotype, what is the relative expression of different markers in this particular cell of in this particular area. So basically you just put a query gate and then overlay it onto the concatenated file, and then again you can create a multi-graph overlay which will reveal all the different measured parameters in your experiment and their relative expression with the different color coding. So here you can see the background is gray and this is pink. So you can see the pink ones are expressing like high CD56, CD94 and stuff like that. And if you have gated different patient, let's say different conditions, experimental condition, you can overlay them within your query gate to know whether any of these conditions or patients in this case are differentially expressing this particular cell population. For example, I can see patient 1, PID 1 is highly overexpressing this particular cell type, whatever this is overexpressed in patient 1 and almost depleted in patient 3. And rest all like PID 2, patient 2 is also very less, and 4 and 5 are more or less similar. So now if you're really interested what phenotype this particular area has, you can have a look by using this multigraph overlay. And the beauty and the ease of using this query gate, once you make a query gate, now you can move it around on your population, on your total this t-SNE space, and and this will automatically update these visuals. So it will automatically update, and every time you move it around and wherever you are land in the new location, you can see the differential marker expression. So I think I'm quite running within time, I guess. So this is when you have all those different conditions, like see the flow sum clusters that you are getting out of this whole exercises, 31 clusters you're getting, and different level of expression of each of the markers, like a heat map. And also you have added as columns the different experimental conditions. So now, earlier it used to be you have to export the individual clusters as FCS files, then load them back into the workspace. But now with help of Cluster Explorer, you don't have to do that. Explorer can do it in one shot. You can just, there's an option, you can just click on that, and it can derive all those MFI and frequency for you in a CSV format, comma separated value, which you can open in an Excel. So this is Cluster Explorer that I was telling, and today I plan to show you as well. So what it does basically, it is interactive between the dimensionality reduction and the clustering tools. So basically it shows you with the principal component line plot, where it shows the relative expression of all the different markers in your panel across the different clusters. So CCR7, for example, you can, I'm not sure whether you can able to see that. CCR7, CD16, all these markers are getting, some are elevated, some are depleted, some are downregulated. All those things are happening across the different clusters that you have got here through the phenograph in this example. And you can see in this heat map, the number of clusters are shown as different rows, and the columns are different antigens. So basically this heat map is very much informative. You can really have a quick glance on it and identify the particular antigen which is getting upregulated in certain clusters. And you can really gate different antigens and then compare them in this particular plot, either here or here. You can select multiple, there are and or gate options are there. You can select multiple clusters and compare the different antigen expression across in these clusters. So we plan to show that in today's session. So basically this is the last slide that I have for today. So plugins facilitate interaction with bioinformatics program environment and use of algorithms. For a bench scientist, even that person is not required to have a vast bioinformatics experience. It's so easy once you getting used to this workflow, like with your data set. Try using with some simple data, maybe not should not have be like a 20 color data, maybe 6-8 color parameter data is also fine when you're starting to do practice with your and getting used to using this informatics tool. And it is really a matter of practice and getting used to while you are in this different plugins. And employing plugin tools is the proper workflow, allows for discovery of complex potential novel populations. Because what I told at the very beginning is that you cannot really derive novel populations if there are such populations in your data hidden. You cannot do it with manual traditional gating with your available literature knowledge or what we have, because that never reveals. But these computationally derived like algorithms, they can help you out in getting there and finding those novel phenotypes. So I end my presentation here, and then what I would like to, what is, we have some time now. I think, Ankita, we can have some questions, and then we can probably go for, I can go for the live demo.
Sure, Amlan. The question one is live now, Amlan.
Okay, so the participants can take part in the quiz and submit the answers. I think people have started, some have started responding. So the first question I believe is, identify the true statement from the following. Phenograph requires number of clusters input from user FlowSum requires a number of clusters input from user. Both are correct. None of the above are correct.
We have got the certain
Sorry, I was on mute for a while. I'm so sorry.
yeah, no, we lost you.
Okay. Okay. Yeah, Peter, you can continue.
Yeah. So the questions, I think we've already got 18 participants who have responded, and the number is going on. Yeah, 20 now.
Okay. I think, I mean, I can discuss it. I mean, while I was in my last few slides, I told that when you are using this flow sum, um, it really asks you to a user to input the number of clusters, meta clusters. So the answer B would be correct.
Yeah, almost 77% of the people said B. Yeah, good.
Great.
Yeah, thanks, Amlan. Ankita, you want to get the next question?
Yes, sir, it's there. So we have Nirupama. She's wrote something. Sir, Peter, can you please ask her for me? Okay.
Yeah, so
So I want
Dr.
to ask
Nirupama
her.
has a question, Amlan. She
Yeah.
need to ask about the reduction strategy. She wants to understand the reduction strategy. Dr. Nirupama, Ankita, can you unmute Dr. Nirupama?
Sure, I can do that, sir. Just a minute. Till people are responding for
Okay.
the
Okay,
question,
Dr.
I
Nirupama,
will.
will unmute you. You could then ask a question, okay? Because probably that little line might not understand the question well. So it could be Tim or even Amlan who could answer your question.
So we are at the
I'm
second question.
I'm here, but I'm not sure what the question is about a
Yeah,
reduction strategy.
yes. Tim, that's the reason I've asked Ankita to unmute Dr. Nirupama, and she could clarify the question. She can ask the question live.
Yeah. So, Nirupama ma'am, you can now, ask your question. You can unmute yourself and ask the question.
Tim, I wanted to ask the how to do the reduction, because in the one of the slide you have shown that after the CD45 you can reduce the 15 and then 3 and then 4. In the last you can only see the CD8. So I was not able to understand that how we can do that reduction.
Oh, yeah. So
Tim,
with that particular slide, and if you wanted to put it up there, Amlan, we can talk about that. The point of that slide is that you can create a dimensionally reduced space, a dimensionally reduced data embedding using tsne, UMAP, TriMap, or even embed sum at any level within your gating hierarchy. So depending on your experimental question, you may want to look at all of the lineages in your system. And in this
Yes.
slide, it's showing the very first is just we gate down to clean CD45 positive immune cells, and then run tsne and overlay our manually gated populations. And you can see that the granulocytes there in a whole blood sample take up a large portion of the space in that dimensionally reduced embedding. So if your question is not about the grams, if your question is really about CD8 cells, you may want to use a gating strategy first that gets you down to the lineage of interest where your question is, and then dimensionally reduce at that level so that you can spread out the populations and see the peaks and valleys, which represent different combinatorial expression states of the cells. If you are always looking zoomed out at the entire system of whole blood or PBMC or whatever, you're going to look at all the lineages, but the detail within a lineage won't be there. And so you can gate down to any level in your hierarchy and apply that that strategy of dimensionality reduction and clustering to that subset to answer your question.
Okay. So it means that I have to only get the CD8 T cells and then go to the reduction, dimensionally reduction strategy. to see the.
Correct. Yeah. And that's part of the cleanup process, right? So you can gate down to as clean or clear a population as you need to get the resolution within a lineage or a set of lineages. You don't have to dimensionally reduce at the level of all cells or at the level of CD45 positive events. You can go down and use a lot of high parameter panel design is using dump gates to get rid of a whole bunch of lineages. Like you could put CD14, 19, 20, and 15 all in the same color and just gate all those cells out to get rid of them, and then focus in on your analysis and use the parameters to get more information out of a specific subset.
Okay. So in this slide was looking like this, that after doing the CD45, you can reduce this to CD15.
Yeah,
You mean to say that with the TISNI, we can find out the granular size, and then CD19, 20, and then 56 and 4. And but in the ultimately, you are if pointing out the CD8 T cells, then you can finally do the TISNI on the CD8.
correct. And this is just sequential gating, in this case, removing CD15 to get rid of the grands, and then running the dimensionality reduction at that level, removing CD14 on top of that, so you get rid of the monocytes, removing 1920 events, so you get rid of the B cells. And we're just essentially stepping our way down into the gating of our lineages to get at the CD8s. And at each level, you can run dimensionality reduction to get this view of your data. The question becomes, what's your experimental aims, your hypothesis, and what do you need to focus on for answering that? And sometimes it's looking at everything in your system, and sometimes it's gating out a bunch of lineages so that you can focus in on the T cells or the B cells and answer your specific question. Now, Amlan, can you go to the next slide there real quick with the color maps?
Yes.
So the question was about HLA-DR expression in the monocytes up there, and I admit that the DR expression is not super high in those cells in all the areas of the monocyte, but it may just be a scaling of the color axis parameter that doesn't give us the nice, the brighter color red. And so I'm not going to go into it. I think you can use other markers in there. Maybe this experiment, the HLA-DR staining wasn't that great, but you can see CD14 and 11C and 13 there in the monocytes, which is that island up at the top. And if you follow a monocyte island out into the peninsula that's way up at the top of this plot, you'll notice that you get really high CD45 RA as well as CD16 expression. And so you can see the monocytes there, and then the patrollers that actually have lower CD14, lower HLA-DR, but express CD16 and 45 RA. And the point here that I'm just going to make is that when these maps look very unfamiliar when you first start looking at them, but when you start doing this dimensionality reduction on different data sets, you'll find that the shapes of the islands actually start to become a little familiar, and you can tell, oh, those are my B cells or those are my monocytes, just in the way they look. If you're doing dimensionality reduction with panels of reagents that have similar parameters in them, so it'll become more and more familiar as you get to look at it. Yeah, well, 123s are a little island over on the left there, and I do see that too. So, I mean, that's a deficiency in the data. We'd have to go and look at the HLA-DR staining in this panel and dig into that if that were the case.
Okay.
Okay.
I think there's a question from Priyanka. Dr. Priyanka, what is your query? I've not got your query, and can you, Ankita, can you help Priyanka?
I think, sir, she's already put in her question, right, earlier, wherein Tim had helped.
Okay, so then, no, that was Preeti, I believe. I'm not
That was
sure.
Preeti. Yeah. After that, Priyanka asked why were monos not showing HLA-DR. So she's referring to a particular slide.
Oh, Okay,
Yeah.
okay, okay. So I think that was already being discussed with Tim. So
Yes,
Priyanka,
I
I think
think.
that's already being discussed. So I think it's fine.
So
And there is a
it
suggestion that somebody wants to do it practical. I think when we get live into the software, we are going to help you out with that. Okay. So is this the third question now, Ankita?
I believe it is the second one.
Or second, everybody's answered perfectly. Looks like, right?
Okay.
Uh, yeah.
The answer would be
Second.
true.
Yeah, so I think almost 100% of the people have said it's true. So I've already seen that. Yes.
now I've shared the third question as well, sir, because we were having discussion. Yeah, it's live now.
O- Okay. And is that the last question before we move back?
Right, sir.
Yeah. Okay. Okay. So this is the third question of the poll. It's identify the correct sequence for a high dimensional discovery workflow in a FlowJo. Is it cleanup dimension reduce, concatenate, or concatenate dimension reduce and cleanup, or concatenate cleanup dimension reduce, or cleanup concatenate dimension reduce? So you have four choices and three sequences. So you have to give the correct sequence. Yeah, you can just choose the option and submit. You need not type it actually. And 95% of the people are saying it's the last option. So over to you, Amlan.
Yes,
Amlan,
it
you
is the last option.
tell about that. And just one announcement before we move on to the session. So, guys, we have put in, and I will again put the attendance link in the chat window. We just have 38 people who have filled the form, and we have a total of close to 80 attendees. So, we request all of you to type in your name and organization. That's it, what the form asks, and that's purely for attendance. Thank you.
Okay, so Ankita, I think the rest of the questions we can have in the second break,
Yes,
right?
Amlan. Yeah, right.
Okay. So now what I will do is I will again pull up my screen and then uh, we'll try to. Can you all see it?
Yes, it's come up, Amlan. I
Okay,
can see the FlowJo
great.
software.
Okay, okay. So what I will try to do is now, because all of the participants, I mean here who got a license from FlowJo or who are attending this course and has a FlowJo, they can use this assignment that we'll send it out at the end of today's session. So there you'll have 5 FCS files and a workspace template which you can use for analysis. All the gating hierarchy and everything has been done so that you don't spend time on the initial level of analysis, and you can directly jump into the high dimensional plugins by using those instructions that also will be provided along with a set of like a figure manual, so that every step has a reference figure what you can relate to, that whether you are at the correct way or not. So what I will do is I have those things ready. I'll just quickly open my. So this is the workspace template file which I'm dragging up here. Okay, yes, maybe the version is different. You don't need this. And then these are the 5 FCS files that you have. This is basically a 25 color broad immune phenotyping of like broad lineage markers. So I'll select everything and drag and drop to the FlowJo software. So I don't need this anymore, and even minimize it. So what you see here, there are a couple of keywords which currently is not displayed. So what I'll do is I'll go to configure, go to edit columns, and then try and find out those. Yes, these are experimental conditions and patient ID. You can select multiple and add columns and click OK so that they will be displayed. So now you can see you have experimental condition and patient IDs 1, 2, 3, and 4 and 5 for all those individual FCS files, and experimental condition 1 and 2 for two different conditions. So what I will do is I'll just select a particular node of this gating hierarchy. For me, it is the myeloid or lymphoid that I'm interested in. And within that, I want to now first go to so the cleanup, everything has been done on this data set. So what I'm directly
Yeah.
going in is sorry, okay,
Amlan, they want you to maximize your screen. So I'm not
okay.
sure whether you'll be able to do it and still show because screen is a little smaller. Okay,
Does that help?
looks yeah, looks better.
Okay, okay, this is possible. I can go even a little bigger on that. Is this fine? Okay. Peter, can you confirm whether this is better?
I think it's better. Rashmi, I think this is what because he needs a little space at the side to show other things. So that's the reason. Yeah, yeah. So she said it's better. You can go ahead.
Okay, thank you, thank you. So what I will do is you have to select equivalent nodes first in order to group them together. So first you click on any one of that particular FCS files and the particular node of interest, then right click here and then click on
Amlan,
the.
could you explain this gating strategies quickly because you're not doing it, but just show them what are the
Okay,
hierarchies you have created, and then you can proceed. Yeah.
okay, so basically we start with the singlet gating that I showed in the cleanup of that particular slide. So if I
Yeah.
just open that slide deck, so this is the strategy, I believe, this one. So this is the cell of interest, and how we arrive here, we do a singlet gating and then with the forward scatter height and area, and then we do another round of singlet gating forward scatter width versus area, where you select this horizontal gate and within that all the cells are believed to be singlets. Then you do all leukocytes you take together in a gate where you get rid of any debris that might be on the edges. Then you do the CD45 gating where you select all the CD45 positive cells, and now within that you have option for either, I mean, you can cut out that granulocytes by HLA-DR gating, which looks like a stringent gate on the side scatter versus HLA-DR. And this is your myeloid or lymphoid compartment which you are interested in. So that is the gating hierarchy that we used here. So single cells from their singlets, and then leukocytes 45, and then granulocytes we are not interested, and then the myeloid or lymphoid, and within that we have separated different lineages. That's what Dr. Nirupama's questions were. And now if you perform the dimensional reduction on any one of these level, then you are really getting those different outputs. So if you really select CD8 and then do the dimensional reduction output algorithm, then you would really land on the last panel of that particular slide. Okay, I think that should be
Yeah.
answering your question. Okay.
Sure. Sure. That's fine.
So you select the myeloid or lymphoid and then right click and select equivalent nodes. So what is happened is all your 5 FCS files with that myeloid or lymphoid node is getting highlighted. Okay. So now what we do next is you right click here and then concatenate populations. Click on this option, export concatenate populations. Now here, once this dialog opens, go to the concatenate button, and now you have to specify a location for the output concatenated file to be saved. For me, it is the desktop. You can choose any particular directory where you're interested to keep your concatenated file. Now, this is one important thing that which I want to highlight. So in my workflow, I showed something as downsampling, where it makes sure that equal number of events are going into the downstream analysis. So I mean, when you have this particular version 10.7 of FlowJo, you don't have to do downsampling separately. You can just click on this, include no more than whatever it's showing 57,418, which means in particularly any one of your this node is having this number of cells. So this is the maximum number of cells that you can include for a downstream analysis. I'll just quickly confirm and show you that. I'll just cancel this for a while, and you can see this myeloid or lymphoid node is having 82,000 for the first one. For the second one, it is having 76,000. For the third one, it is 92,000. For the fourth one, it is 57,108. 18, and the fifth one is 71. So the least is this one among all the 5 files. So whenever you are going for the downstream analysis, you can have maximum this number of events from all across your all 5 files. Make sense? So what we do is we just perform that downsampling in that concatenation option. Select that, go to concatenate, and then you select the destination, which I've already done, and then I'll include no more than, for this demonstration purpose, I'll select 10,000, but you can go up to this 57,000 what you've just seen, but it will take a long time anyway. 10,000 per file is 50,000 total. That will also take quite a lot of time for me. So depends on your system configuration and the version of FlowJo that you're using. So 10,000 I'm restricting this to, and then in the advanced option I'll just put a suffix which will come in your derived file. So maybe I'll just put today, whatever you can put here. And this is very important because when you are concatenating the file, we always encourage you to go to this additional parameters and choose the newly derived keyword based parameters that you have just created. So if you scroll down and look for the option that you created, you can find it out, the experimental condition, this one, and you can press control, press down and select multiple. Here multiple you can select. So for me, I'm just selecting experimental condition and patient ID, and then click OK. So you can see additionally these two parameters will also be taken into the concatenated file. Now when you click concatenate, it will ask you that where to open that file. So for me, you can open it in a new workspace or in the existing workspace. So I'll open in the existing workspace and click close and then cancel. So now if you go down, you can see there is a new file which is concat_1_today. Today is the suffix that I've just added, just in order to identify this particular file. And you can see it has 50,000 because from 5 files I have taken 10,000 each. So 50,000 cells are here in this particular concatenated file. So next comes is the dimensionality reduction. So once you have that, so you have to select this particular concatenated file and then go to the workspace, and you can see there's the native button for t-SNE. So if you click on that t-SNE, you'll see this dialog box opens, and which will actually give you different options for choosing what all parameters you want to be the t-SNE to be performed on. So for me, it is a 25 color immunophenotyping panel. So I would go ahead and choose all those measured 25 parameters by selecting each of them. And then I will, there are a bunch of options below. So if you can see here, the by default option is the opt-SNE, which is there. The reference one is the Anna Balkinia's paper. And then iterations is, number of times the algorithm works through your data before it takes for a halt. Perplexity is related to the nearest neighbors, and learning rate is like how fast you're going to achieve the desired result. So more on these different conditions, Tim will be taking you through in the next session. Today's after I end with the live demo, so Tim will be explaining more on these options, what it means. And then here also you have options for k nearest neighbor algorithm. One is the exact vantage point tree, and another one is the approximate random projection forest. And also, if you want to gradient algorithm, one option is Barnes Hut, the traditional one, and the other one is FitSNE. So if you want to really have fast output from your experimental data, you should go ahead with the approximate or random projection forest, and also you should use the FitSNE. But even if you are not sure how your data would look like, I would encourage you to you can just select the default parameters, what is there given, and then you select the measured parameters, all the fluorochromes. all the compensated parameters that you used for the experiment, and then click on run. So as you can see, this dialog box on the top right corner has appeared, which is showing like 1,000 iterations, if you remember. That is the input that I have given, and it will take time to complete that action. So it will go from 10, 20, 30, all those iterations it will complete, and this progress bar will show you when the t-SNE gets completed. So okay. Yeah, so you can see iteration is just started. So in the meantime, if you have some questions, we can take it. So if Peter, you can see some questions in the chat box. You can just read it out to us.
Yeah,
Maybe I.
Amlan, so most of the questions were coming on the R, challenges which you are facing. So I have
Mhm.
replied that we will work it out during the breakout sessions. Okay.
Okay.
Uh,
So
and attend.
people have got the license, and it seems that they've started. Tried to install the R, right?
Yes, looks like.
Mm.
But yeah, so I do not know how many have received it because I have seen a lot of licenses being sent and still open. They have not logged in, but I can check on it later.
Mm-hmm. Okay. So for today's purpose,
So,
let us. Yes.
Mandar and Nirupama have raised their hand. Do you want to ask them to put their questions in the chat, or how do we want to go about it?
No, I think
Yes.
they can ask the question now.
So,
We have
should I
some
ask?
time to.
Okay, okay. So, first I'm starting with Mandar, okay? Mandar, you can
Okay.
ask your question. Mandar, do you want to ask the question or you want to type in the chat? You can unmute yourself. Okay.
This is Tim and Kita. Can I answer that question?
Sure, sure,
So I
Tim.
Okay.
started
Sure,
typing
sure,
into
sure.
the chat, and the first question was, well, at least in the chat, the box that is a square badge next to the sample names looks empty, and that is because these samples were actually exported as compensated data. So they don't come with a matrix, but they are actually showing just the compensated data, not the raw data. And then the second question was, and you can do that from the export concatenate platform as an option. And so for this, the simplicity of this example, we exported only compensated data. The other point here is, do you select parameters which have been used for cleanup as well? And that is really your option. For one thing, you may have gated out lineages that you aren't interested in, and in that case, you may want to leave those parameters out of the algorithm because then they won't be considered during the dimensionality reduction and embedding. But if you say gate for CD45 positive events, there's a lot of information in different immune subsets with respect to how much CD45 they express. And so you may want to include that one because there's discriminating information. There's variation between the cells along that parameter, even though you've already gated for positive events. Similarly, if you think about forward scatter or side scatter, there's a lot of information in scatter profiles. If you're looking at monocytes and lymphocytes together, right, there's differences in their side scatter. If you're only looking at lymphocytes, then there's not a whole lot of difference there. So if you're looking at all the different lineages and there's variation across those cells in their scatter profile, then including the scatter parameters is worthwhile because it adds discriminating information. If you're looking at just one population and there's no difference in the scatter profiles in those cells, then maybe you do not want to include the scatter parameters during calculation. So that's really your option. And you just have to think about it with respect to which parameters are meaningful in this experiment and which ones do I want to include to separate the cells visually with this dimensionality reduction approach. I hope that helps.
Yeah, I think thanks for that. Mandar, you still have any other question? Because you were asking for a unmute. You could have unmute yourself and ask the question. I think Ankita has unmuted you. She's given you the option.
Yeah, also Nirupama has again asked, so I am allowing her also to unmute.
Okay.
I just wanted to ask Peter that what do you
Yeah.
mean by our license? That it's a kind of a that we have to purchase the R.
No, no, no. The links were given Dr. Nirupama last time in the session. Those links are there and it was explained and we could send you the session. You don't have to purchase it. You have to just download it.
Because I didn't get the last time the link, so
Okay.
that's the
Okay.
reason. If you
Okay.
can after the you can send me. Thank you
Sure,
so
sure,
much.
sure.
Thank you.
Okay. Okay. Okay, Dr. Nirupama.
So takes, as you can see, takes quite some time.
As this goes through and calculates,
Yeah.
and this is the challenge with these algorithms, is that they do take time, so they don't lend, well to live demonstration as it runs.
Yes.
But the question I believe was, when do you use Flow AI or Flow Clean? And the answer is on that, the root sample, when you first bring it in, you should run Flow AI or Flow Clean to clean up that sample and get rid of any clogs or bad events that are different across the collection. And that is just an optional cleanup step that should be performed at the level of the sample when you first bring it in, and then you work downstream of the good events gate that comes out of that. And when I show my screen, I will show you an example in FlowJo of where I've used Flow AI prior to the concatenation step. So it is part of the cleanup. You do it before you're gating, and you work with the good events that come out of that as the clean sample. In terms of discussing how to use it, I don't think we're going to go. I don't even believe I have a slide showing specifically the user interface. But in all of these plugins, when you spin up, when you select them from the drop-down plugins menu, they'll show a graphical user interface. And with plugins like Flow AI and TSNE and UMAP, we do have links to the papers right there, so you can actually pull up the paper, and I would highly recommend reading that before employing Flow AI, because then you'll understand. And Kate's going to show it, how to install it in terms of our setup here in a little bit. But it's always worthwhile to read about the options before you start clicking all of the different buttons in there to turn on and off options within these user interfaces. Generally, we've chosen settings that work on most data quite well. And so the defaults are usually my go-to when I demonstrate and employ these for real purposes. So don't get overwhelmed with all of the different options within those plugin UIs. It's worth just giving it a go and making sure you get an output, and you'll find that, if you apply Flow AI to a really cloggy sample, then you'll see it clean up quite well.
Okay, I think the FlowJo clusters are there. We're able to get some output from the dimensionality reduction algorithm, TSNE. So if I continue with the session, so what you can see on the screen is the different visualization of the dimensionality reduction algorithm TSNE that we have used. What I would recommend is, as you can see, the clusters, few of the clusters are touching on the axis, so you can just click on this T button, transform, and then go to customize axis, and then just select this just to have some space on either side, and then select both of the TSNE parameters and then apply, so that you can see now you're well off quite at the center of all those different continents. And now the first thing that I would like to do is you'll click on the y-axis to have the histogram display, and then try to see the different experimental conditions that we created by clicking on the patient ID And these are the 5 different patients which have contributed to this concatenated file, 10,000 cells from each. So now I will use the range gate, which is a standard an option here, to gate about these individual conditions. So I'll click on that and go selecting each one of them like this. This is my patient 1, so I'll just click 1, which is obviously 20% of the total, and then go to number 2, then select third one, go for the fourth, and there you go. Okay, so these are the 5 different conditions. As you can see here, they'll also be shown here as 1, 2, 3, 4, and 5. Each is having 20% and 10,000, because we have restricted while doing the concatenation that maximum 10,000 cells will be taken from each of the FCS files. So now we'll again go back to this t-SNE, and then I'll show you how to use the third parameter, color mapping. So as you can see here, this is the dimensionally reduced fewer dimension space, and where you have your data embedded, the high dimensional data in the low dimension space, and these two are the newly derived parameters, t-SNE X and t-SNE Y, or t-SNE 1 and 2. So on that, if you want to see the expression of CD127, let's say, you can just click in this box, color axis, and you're able to see the different gradient of expression of this particular marker. And in this graph window, you can see one marker at a time. So if you click on this drop down menu and select any other, like HLA-DR and CD56, CD45RA, all these, you can select one after another. But what you can do is you can really have a look at all of them together. So for that, I'll just open this layout editor, maybe create a fresh layout, and then I will right click here on this graph window and copy it to the layout editor. So it has come here, and within this layout editor, I'll just right click on this and then go to the last option, make multigraph overlay, and within that the last option is multigraph color mapping. So it will ask you which all parameters you want. For this demo, I will select all those 25 again, just to have a better understanding. And then click on the choose selected parameters. It will take a while, and then it will give you the output of 25 different marker expression across that dimensionally reduced space. So now I'll just make this, enlarge this a little bit, and you can see like all these, this is really help you to orient yourself with the data. That as I was explaining, like this is your CD19, this is your CD4s, and maybe this is your CD8 population. Like now you can see this is the global structure which t-SNE sometimes fails to preserve. So CD8, you can see it's here as well as here. Ideally, CD8 should be located in one specific island, but see it is broken up into 2 different islands. So similarly, for the other markers also, you can have a quick look of the expression across the different like concatenated file in the visualization area of all these different markers. So this is one way of looking into it. And the next is what I would like to do is I can create something called a query gate, which is also helping us to go deep inside any particular area of this population, and to know what exactly is happening in terms of marker expression. So for that, what you have to do is simply you have to let's say you have to select any one of the gating tools that you can use. I would use this pencil, which is quite a smooth one. So you can just select any area which you want to see details of. So you can just encircle it, and for me I'll just put the name as query. So once you create that query gate, which is capturing 8.4% of the total, you can see that is also getting highlighted here. Okay, so the total, this particular file has 50,000 cells, so 8% of which is around 4,000, so which is exactly matching. So 4,000 cells are there within this particular area. Now if you have a real quick look at the different conditions that you created, like all the different patients, so what I'll do is I'll select each of them, 1, 2, 3, 4 and 5, and then just drag and drop on the query gate. So now what you'll end up with is having within those 4,217 cells, how is the distribution of all the different 5 patients? And you can clearly see patient 3 is having like more number of cells compared to the others. Like this particular patient is contributing to a large extent of this particular population compared to the rest. So this also gives you an idea. And from the color map, if I go back again to the color map, you can see like this particular island is expressing like CD19. Maybe the B cells are having like a high expression, high elevation in that patient tree, and this could be related to a disease phenotype or something like that. That would be the researcher's call. So this is a very quick thing that you can do on your data. Next thing, what you can really, stretch and do on the layout editor is can just bring a fresh layout editor, and then I'll just right click, copy to layout editor, and within that I'll just put the query gate, select it, and try to overlay it on this. Okay, so I'll just select query gate and put on top of this. So once you do that, you'll see it is okay. Once you do that overlay your query population on the total concatenated, you'll see there is a difference in the color mapping. So this query population is the population of interest, and rest all is marked in a different color. So you can change that even. So if you want a bright color, for example, you can click here and then select a maybe brighter option. And now what I'm really interested is just to have a look at the different markers expression which is going on within this query population. You can just click or right click here and go to the last option, make multigraph overlay, and select histograms. Now once you do that, you see a bunch of histograms are shown here, maybe all of which are not required. So you again select that panel of histograms, then right click and go to the last option, which is select multiple parameters. Now when you have select multiple parameters, you can just go with the 25, not these things. So these are may not be of interest to you. So only the measured parameters in your experiment, like all those 25 fluorochromes which you have stained with. So you select that and click on the choose selected parameters. So what happens is you can now enlarge this in order to see what is the relative expression and how this thing is changing. As because we know from the heat map also, this is more expressing B cell markers, which is again getting confirmed here. See CD19, IgD. Okay, and like you can see, it's clearly negative for CD3. CD3, which is in red, it is expressed somewhere else, not in the query population. So this will also help you to understand and go deeper inside this population. And once you start moving your query gate in this graph window, you'll see this visuals are getting automatically updated. So I click here and then I move my query gate and move it here. You can see, so this is maybe 45 RA high, this is CD3, this is not CD4, this is CD8 expressing cells, but again CD8 is also expressed in the rest of the populations, and we know this is coming from somewhere here. This island also has some CD8 expression, that's why this is coming here as well. So this is how you can just make query gate once for all and then move it around to your area of interest, and this will automatically keep updating, and you can have those visuals. So this is again one part that was very important, that these all applications you can do by simply doing the dimensionality reduction. Now we will do a clustering algorithm, which is X shift, which does not require use of R. So I will minimize this. What I will do is I will go to this particular concatenated file, select it, just highlight it, and then select, go to my workspace. No, this is, I'm already in workspace. Okay, why does it ask for, anyway, I'll just maybe, okay. So I will select this concatenated file, highlight it, and go to plugins and select on X shift. Okay. So once you select X shift, what I'll do is again select all those parameters, the 25 fluorochromes that I've used for this experiment, and then do not, you cannot change this numbers. Okay, this 35 number, this is given by the software, you cannot change it, and then just click OK, and then you'll see it is calculating X shift. Okay. Oops. Let's close it. Think that should work. So it is pretty fast. Xshift doesn't take much time, and it will give you the output as some number of specific clusters it will show. Okay, so here you go. So we have got total of, these are the different clusters which are, I mean, given as specific numbers 1 through, we have total 37 clusters, which is the output which has come. So I mean, in my hands, it comes with these set of, I mean, plugins that I'm using, the versions. I'm really getting 37 cluster output from this using Xshift. Now the interesting thing is we will now spin up Cluster Explorer and try to see all of this together, like Xshift derived clusters in the dimensionally reduced t-SNE space, and try to see the different expression of all those markers and heat map stuff like that. So for that, I'll have to again select that concat file and then go to plugins and select, okay. Oh, I'm not having that, maybe, okay, the version. So I have a couple of FlowJo versions in my PC. So I'll just quickly save this and open in the updated version in which I have this FlowJo. I'll just save it and then close it. Yes, just give me a moment. So FlowJo, I'll just have in 7.1, and then I can open that and show the Cluster Explorer to you guys. Yes, there you are. So once you have this open, so just I'll quickly click here and then open the saved workspace file, which is this one, and then we have everything updated here. Yes, all the 37 clusters you can see here. I'll doing the Cluster Explorer, I'll just select this file, which is the concatenated file, and then search for that plugin which I didn't find in the earlier version. So plugins, yes, this is the Cluster Explorer. So I'll click on Cluster Explorer, and here you can see the command which comes is I will use the Xshift derived clusters, whatever clusters I've got through the Xshift, and then I will select those 25 parameters, their expression I want to study, and I will use the t-SNE x and y, all those derived dimensions, t-SNE x and t-SNE y, to display those populations. So I will click OK, This ideally should not take much time. You'll see it, I mean, happening in a moment. Yes, there we are. So maybe the visuals are bad, it's taking time. Okay, it has come. So what I can see here is I'll just try to reduce it in order for you to see, and then try to squeeze in everything in the small space that I have. And this is the heat map. So this is a control panel that also comes along. You can try different options like here, which is, I mean, so it would be fun to try different options here. And this is the T-SNE space. So what exactly it means is once you click Cluster Explorer and use the 37 or whatever, yes, 37 number of clusters that you derived from X shift, and also display all those cluster in the T-SNE X and Y, T-SNE space, the dimensionally reduced space. So all these different colors actually designate one individual cluster. Okay, so I'll start from the first window that we got here. So basically what this tells is it shows you the different clusters that you have derived, 37 clusters, and this is the number of cells. Event is number of cell. What clearly you can see, some of the clusters are having unusually high number of cells. So you can select like one of them. Let's say this one I select. So automatically you see this particular cluster, which is in green, it is getting highlighted. So this line graph tells, this is the principal component line graph, which tells you the relative expression of these level of the different parameters. I mean the different antigens. And if you study this particular island or this cluster, you can see the expression of CD45 is high, it is having CD14 high, CD11C is high. Okay, if you want to compare and study multiple populations together, you can just click control and select another one. Okay, and you see as you go along and select multiple populations here in this plot, they are getting simultaneously highlighted, and you can really look into the different levels of expression of these markers, and they are also getting highlighted here. And if you now come to, because this is cluster number 27 and cluster 33, now if you want to really see the expression in a heat map, so because you selected all these two clusters, you can see them getting highlighted here. So cluster 27 and cluster 33. So all the rows in this heat map are the different clusters, and the columns are different markers, I mean the antigens. So you can expand it just in order to, if you have a big screen, you can just get it right away. You don't have to look for expanding. So you can see CD45 is high, CD27 is high in this cluster. Okay, and stuff like that. So, and this is the intensity scale where maximum is red, minimum is blue. So there are a bunch of options also. I'll show you. So if you want to go back to the original one, so you just have to click in this blank space, double click, or here also as well. So if I double click here, it will give you back all those 37 clusters. Okay. Now you can gate different conditions. For example, if let's say CD45RA, if you are interested, you can just press down your left arrow on the mouse and select this particular few of the points that you get here. Okay, and they will highlight few populations here, few clusters where CD45RA expression is high. And then maybe I'm interested in looking at CCR7, which is CD197. So I'll select along with that these two. Okay, so now I'm really left with two clusters. Okay, cluster number 18 and cluster number 28, where these two markers 45RA and 197 are highly expressed. Maybe if, talks about the naive phenotype, because we know the naive cells, this CD45RA and CCR7 are overexpressed, they're double positive. So like that, now if you want to take this visual and put it on, I mean display, so you can just right click here and copy to clipboard, and once it copies here, then you can open the layout editor. So for me, just open a layout editor, and then maybe a new layout I'll create, and there you can just right click and paste it. So you can see CD45RA and CCR7 are getting highly expressed in cluster number 18 and 28. Okay, now if you really want to know what is there in 18 and 28, let us look into the heat map. So in the heat map, I can see cluster 18 and 28 are highlighted, and I can clearly see these are CD8 positive cells. Okay, so they are having high expression of CD8. So which means, I mean, you can really I mean, explore a lot in this particular area. I mean, either choosing different clusters from here in this frequency plot, or you can select from expression of different markers which you are interested in, and then select them and look at the heat map. Now the last thing probably I would like to show you is how you can really you can as well click different clusters from here also. Like if I select on this cluster, I just highlight this and press down the control and select maybe this. Okay, so whatever number of clusters that you are clicking, they are getting highlighted, and the subsequent phenotypes are also getting expressed in this heat map. Okay, so these 4 clusters I have clicked and they have shown up here. And as well, you can at the same time look at the principal component line plot of, and here also these 4 clusters are highlighted. So these are very much interactive. This space is very interactive, where you really dig deeper and find a lot more information from your all those clusters derived through XShift, and you can really look them live in this t-SNE space. So maybe last thing which I want to show is you can derive this if you just click on this menu option here, and then there are like you can copy this image, or you can copy content. So copy content is if you click on copy content and then paste it in an Excel, it will open in a comma separated value format where you have all the MFIs and frequencies. So earlier you have to individually export these XShift derived population as FCS files and load them back in the workspace, then add those MFIs and then again put it back in an Excel. But now it is like from Cluster Explorer, you can do it in one shot. So the interesting thing, last thing that I want to show is you can click this option, add nodes as columns, and then it will ask you which all nodes you want to see. So within the concatenated file, we have separated all the 5 individuals as patient 1, 2, 3, 4, 5. You just select 1, 2, 3, 4, and 5, and now select the new columns. So what it does is on the side of what you already had, all these 37 clusters and their expression of different markers, you have now the 5 individual patients, and from them, so just keep in mind the color code has changed. So now yellow means in this new heat map, the yellow means highest and the blue means lowest. So now you can see the distribution of these clusters across the experimental condition. For example, if you notice that patient number 2 is highly contributing to these, I mean, it is highly expressing cluster number 19 and 20. Okay, so 19 and 20 are very high in patient 2. For example, again, if you want to go back and study other clusters as well, so you can again click back and then you are here, and what you can do is this will really help you to understand the expression of the different markers across the different experimental conditions. And this is, I mean, I would say this is a huge advanced tool and helps you to, have a quick look at your data and go as deep as you want. So I think I am quite okay with what I plan to show today. Like one dimensionality reduction algorithm, which is t-SNE, one clustering algorithm, which is XShift, and the last one where you mix and match everything and do a lot of deep study, which is the Cluster Explorer. So maybe we can have the second round of questions for the quiz, and then we can Tim can take it away from there.
Sounds good to me.
Thank you.
Sharing the first question, Amlan. You want all the 3 questions, right, Amlan?
Yes.
Okay. Just a minute. The first question is live now, Amlan. And Tim has been answering so many queries everybody's having. Thank you so much, Tim.
That's wonderful. Thank you so much, Tim.
So the first question is live, and I'm seeing raise of hands as well. So we'll take this simultaneously. Yeah. So the first question is live, Amlan.
Yes, people have started answering the question as well.
Okay, sorry, I was on mute. I mean, this is a
Yeah,
stupid thing
so
sometimes. So, I think the question is pretty clear that Disney does a better job in preserving the global structure, true or false? So, are we getting answers, Peter?
yeah, I think 96% of the people have said it's false, and
Yes, that's
that's almost
correct.
like 30 participants have said it's false. Yeah,
Okay, great. So we can have the next one, maybe.
yeah. Ankita, next one.
It's live, sir. Yeah.
Okay. Yes, the question is which of the below can add sample level metadata useful for evaluating multiple experimental conditions, time points in the merged file. So the options are query gate, downsample, keyword, or concatenation. Yes, and the keyword is what is the option mostly people are going for it. Yes,
Okay.
and some are even talking about concatenation. So I think we've got 23 respondents and it's going on increasing. So let's see. Yeah, so majority of them are saying keyword, almost 73% of the
Yes.
of
So
30
that is the correct
respondents.
answer. So when you are really putting keywords, you are just having a lot of metadata which you can really gate apart later once you are done with the clustering and stuff like that. So keyword would be the correct answer.
Amlan, you've done a good job.
Thank
Yeah,
you.
he's
Yeah,
great.
I
yes.
think we have
Next,
the last
the
question
last
now.
question. Yeah, identify the correct statement from the following. Environment planning. A second option, downsample does not normalize the number of events contributed. Third option, hyperfinder is used to solve derived populations. And the last option is both A and C.
So we have two raise of hands. So one is by Lindsay. Should I unmute her?
Yeah, sure.
Yeah. Lindsay, you can unmute yourself and go ahead with your question. And then we have Nirupama. So I have allowed Nirupama as well. So after Lindsay, Nirupama, you can go ahead with your question. Lindsay.
Ma'am, sorry for the inconvenience that was done by mistake.
Oh, okay, okay.
Okay.
Yeah, then
No
over to Dr.
worries.
Nirupama.
Yeah.
Dr. Nirupama, you can
Dr.
ask
Nirupama.
your question.
Yeah, my question is, what are the important plugins we should download it? Like earlier, we were using only this that FlowZone and the Disney, and but now and the UMAP certainly, but the X and the others, I was not aware. So can you please list out that what plugins we really needed for the deep analysis?
Okay, Tim, you could answer this question.
Yeah,
I think
sure.
there's no limit, but then you can still suggest.
Well,
Yeah.
I was kind of typing it into the chat as well. So
Uh,
in my opinion, TSNE is already native, right? So you can just run it, it'll work, there's no problem with that. UMAP is a downloadable plugin, and so if you want to compare dimensionality reduction algorithms, UMAP and TriMap are both good options to TSNE. So I would go ahead and get those if you're planning on employing these methods. In terms of clustering algorithms, FlowSum has become very popular because it also produces some visual representations of the data, and I'll go into that. XShift and Phenograph are the other two clustering tools that I would recommend and I think have a really good place in this workflow. For cleaning up data, FlowAI is my preference over FlowClean. And of course, the Cluster Explorer is what you're going to use to put all of that together, looking at the clusters within the dimensionally reduced data space that you create, and being able to see the phenotypes and pull out all the measurements of central tendency, the medians of all of those populations for each channel. So you can use any combination of these, but those are the essential ones. I would download UMAP and TriMap for dimensionality reduction, FlowSum, Phenograph, and XShift for clustering, FlowAI for cleanup, and Cluster Explorer to interact with those clusters in a meaningful way.
Thank you so much. Thank you, Tim.
Of course.
Thank you. Thanks, Tim.
Thank you, Tim.
Ankita,
So,
did
yeah.
we get some answers in this last question?
We got a lot of answers, Amlan.
How many of them are D, which is the correct answer?
Right.
Okay, so the correct answer in this question would be D,
Yeah,
because
right.
we have seen that all plugins do not require our statistical environment, which is true. Downsample does not normalize number of events; it does normalize. So this is false. And hyperfinder is the one which you use to computationally derive sort populations. So both A and C are correct.
Yeah.
That
So
means answer D is the correct.
84% of the people did answer option D.
Oh, I'm
Okay.
so happy.
So again, a good job, Amlan. You've been a
Thank you. Thank
fantastic
you all.
teacher.
I mean, this is thank you audience for the patient listening.
Yeah.
Yeah.
So I think we are done with the quiz, and then Tim, you can take it from here.
Yeah, just an announcement, Tim, just before we begin. So we still see that 20 people, around close to 20 people, have not filled in their attendance. So it's a kind request because the same names we will consider for e-certificate. So please click on the link. I have reshared it just now. So please type in your name and organization, not in the chat window, but in the form. So, thank you. Over to you, Tim.
Okay, thank you very much. I was just kind of searching here for a slide that isn't in the deck that I was going to present, but I'll go ahead and get started here, and we'll go back and forth. Just based.
Sorry, Tim. Nirupama ma'am, you've again raised, that was by mistake. Okay, Tim, over to you.
Okay, thank you. So I'm going to share my screen, and we'll go through some slides, and then we'll talk about some of these questions. And there are a lot of questions about things like flow norm and flow AI, and I don't necessarily have, or cytonorm. And I don't have a slide for those, but I can talk about them and give you an example. So let me start by sharing my screen here, I think. Share screen, and there we go. So hopefully you can see that. I'm going to go to my presentation. Can everyone see this presentation slide?
Yes, Tim.
Okay. So yeah, thanks for your time here today, guys. I'm going to go through some of the same information that Amlan went through just to reinforce it, but then I'll pull up FlowJo and kind of talk you through another example of data. The workflow that we're employing here for discovering new populations is really about mining your data thoroughly. I don't think it completely replaces your manual gating for what you know, but it can potentially find complex populations in high dimensional data that are novel, new, and exciting for you to follow up on. And of course, as Amlan went through this workflow already, I'll be succinct here, but we want to clean up the data, at least down to good, clean events. That means cutting out any doublets, getting rid of dead cells and debris, and then potentially gating down to a lineage of interest depending on the scientific question that you're trying to answer with this multicolor panel. And then we're going to take all those individual cleaned up samples and we're going to merge them together using the concatenate step and options therein within FlowJo. And this essentially serves two purposes. One is that you have to put all the data together to get a common dimensionally reduced data space, this T-SNE embedding that we've created here. If you do run T-SNE separately on each one of the individual files, the data is different from each of those samples, right? It could be different patients, it could be different stimulation conditions, and you're going to get a completely different dimensionally reduced data space if you run it each time separately on each file. And so we can't think of dimensionality reduction as gating in the traditional sense. And with gating, we gate one sample and we drop those gates onto all the other samples uniformly, and then we pull out the numbers. Here we need to actually merge the samples together, dimensionally reduce all together, and then we have a system that we can pull apart, again using gating to pull apart your different experimental conditions or even individual samples. So during the concatenation step, if you have different conditions, you want to label them with integer values as keywords and then create new parameters. In this case, I had 4 different tissues and 4 different time points from each tissue. So that gives me 16 total samples that are going into this one dimensionality reduction, giving me this T-SNE continent, which then I can explore with things like query gating, or I can subset out with a set of clustering tools. So in terms of clustering, we have a number of different algorithms that subset or classify these events into distinct populations. And you can visualize those by overlaying the clusters onto your T-SNE space. And then as we've gone through here, with Cluster Explorer, you can get out the median values for all of the clusters for each channel, for each measured parameter, and visualize that, pull out the statistics, or copy this image over for presentation. Now, if we want to separate the samples, we can look at any one of these parameters that we've created based on keyword classifiers. So these are metadata labels that we've given each of the 4 samples from the lymph node, liver, spleen, and blood here. A 1 or a 2, sorry, a 1 or a 2 or a 3 or a 4. And now we can look at those and pull them apart. And Amlan just showed that with individual samples, but here I actually have 4 samples from each tissue that represent 4 different time points underlying these histogram peaks. When you visualize each of those subsets from the different tissues here, you can see that within this common dimensionally reduced data space that you have enrichment of different islands, of different regions. And this is showing visually that these samples are different and they have different cells and they have different phenotypes. And so in the blood here, we've got a bunch of monocytes, some B and some T populations, and the spleen is enriched for B cells and different T cell subsets. The liver over here this is all innate immune cells and CD8, so a bunch of NK-like cells. And then lymph node, we've got B cells and T cells predominantly from the lymph node. And you can figure this out using your color mapping or overlaying your gated populations that you already know. Oh, and I realize I'm not showing my screen, so I might as well show myself for you guys today. Hi, sorry about that. And our goal here is ultimately to say, well, which of these populations, which of these phenotypic states are enriched in one condition and depleted in another, and we can identify those populations as differentially expressed and understand where they come from, which tissue, which group of patients, whether one cluster is there in a disease state and not there in healthy patients, right? That's what we're trying to figure out here. And so this workflow can be employed on any number of samples. It can be just, well, one sample, then in that case you don't need to concatenate and you just have a T-SNE and clustering on top of that. Or it can be a number of samples, and again, the concatenation step is where we can actually merge those samples together and downsample to balance out all of the different the number of events coming from each of these different samples and conditions. Because when you collect different numbers of events, if you just concatenate everything together, well, then some samples will have more events, some will have less. And then when you gate apart these different conditions, you aren't going to have a balanced equation that is 25% of the events coming from each tissue. It's going to depend on how good the sample was and whether you got a lot of events or few events collected on the instrument. And so concatenation normalizes by downsampling during the concatenation process, including no more than a certain number of events. And so each condition is represented equivalently in our system. Now, the plugins here, they continue to expand, and we are adding plugins at a rapid rate, adding features within the plugin so you can do more of the technical options within those algorithms. And, they can be classified into general groups. We've had a bunch of questions about cytonorm and the FlowAI, FlowClean algorithms. Cytonorm is a method for normalizing across multiple runs of data. And the essential thing that you need there is the same sample run every single run. So you want to pick a sample that you have lots of cells from, or maybe make a buffy coat sample where you can make tubes of cells that are frozen, and you can pull that same sample every single week, run it through your assay, and so you have a consistent sample from week to week. And what cytonorm does is it actually runs a flow sum to create populations within your sample, and then it kind of creates these splines and normalizes across your, shifts the data from run to run based on the changes that occur in your constant sample, in your control sample from week to week, and then goes and applies those same changes to your other data, all your individual samples in your cohort. And so cytonorm can be helpful if you have variability, technical variability from run to run. But I want to impress upon you the importance of doing the same assay from week to week, right? Variations in your standard operating procedures, and especially variations in the technician who's actually running the samples, what job they do during that process, can have a huge effect on the batch effects from week to week, even though you're trying to do the exact same assay. And so cytonorm is one solution to that that's come out, and maybe we can pull up a slide next week to demonstrate more technically what cytonorm does. FlowAI and FlowClean, I would just go with FlowAI because it uses a smaller window as it scans, and it has more options available to customize the algorithm for your needs. But it does a really good job of cutting out small clogs and then stitching back together the good data so you actually get as many events as you can downstream that are good, clean events. And so those are my recommendations there. Now, we'll go through and talk about dimensionality reduction and clustering here, and we've kind of touched on Cluster Explorer, and I will point out Hyperfinder and its use in the workflow downstream of our discovery of a new population. But just remember that nobody expects you to become, super high parameter analysts in the first go. So the data that we're giving you is an example data set with 5 patient samples just to get the workflow down and be able to concatenate, dimensionally reduce, and create cluster populations, and then visualize those clusters within the dimensionally reduced data space. I don't believe it's really fruitful to go and try every single method the first time you go. Just pick one. T-SNE is good. UMAP is great. Pick FlowSum if you like the visuals. Otherwise, go with XShift because you don't have to set up R. Those are the type of choices that you make as you start using these tools, and you just have to go for it and just see what your system gives you, what your data looks like using these tools. So once we've cleaned up and concatenated our file, we want to dimensionally reduce the data. And I'm using a different example than one you've seen so far tonight. And I'll try and be consistent. I won't be completely consistent with that example, but when we switch over to the demonstration, I'll use the same example here. And we've talked a couple times tonight about what is global versus local structure. So all dimensionality reduction algorithms do is redistribute your events in this new dimensionally reduced structure, right? 2 parameters, all your data is there. The question is, how do you look at it? Well, one way to approach looking at the data is overlay your manually gated lineages, and you will see what the grouping of events is and how that dimensionality reduction redistributed your events. Now, with T-SNE, as soon as you get to some mid to high parameter experiments, this is an 18 color experiment. You can use T-SNE or UMAP on low dimensional data, mid dimensional data, and very high dimensional data. But when you get to about 15, 18 colors, you may start to see splitting of the lineages into distinct islands. And so you see the B cell population, the major B cell population is down here, but then there's this island that I gated on CD19 and overlaid what I would call B cells, and I have this distinct island that's far away from the other B cells. And this is the problem with T-SNE, is that as you get more complexity, you start getting good local data representation, but the global structure is lost. That is, the relationship between islands isn't clear. And so you can have CD8 positive cell islands in T-SNE all over the place, right? And they are all the same lineage, but T-SNE, the way it's seeded, the way it's initialized, it doesn't keep those events that are similar to each other. It doesn't guarantee that they're going to be close to each other within the dimensionally reduced embedding that you get. But it does work very well to flatten the data so that you can use a query gate, so you can explore this data space with a third parameter heat mapping and such. And so when we start using other methods for dimensionality reduction, these UMAP and TriMAP methods do a better job of keeping the lineages together, and so that events that are of similar phenotype will be presented close to each other within the dimensionally reduced embedding. T-SNE has this problem where it will start to split up the lineages into distinct islands. However, UMAP can also end up with a bunch of clusters that are kind of overlaid on each other, so it's not as clean to just gate a region and say, oh, that is one distinct phenotypic population. T-SNE flattens, and so you can gate an island and say, oh, that group of events is probably related to each other. You just can't make relationships or infer phenotypic relationships between different islands within T-SNE. In UMAP space, you can say, oh, these events are closer to each other, therefore they're probably closer to each other phenotypically. Using both, maybe T-SNE and UMAP is the best way to get a best overall view and look at alternative views, and you choose the representation that best fits what you're trying to tell your audience in your presentation or publication. Now this is another data set. Now I'm going up into 28 color world, right? So from 18 color world with all the lineages represented, this is a pan-immune panel. So we've got NKs and Bs and Ts all marked with their lineages, and then there's some additional subsets downstream. Now this panel focuses in on just T cells, T cell memory subsets, and then downstream effector functions. And so this is actually an OMIP, a high parameter published panel in Cytometry Part A, where the data is publicly available on Flow repository. You can download it yourself and recapitulate this. And these islands are actually not related. Just because they have the same colors, that doesn't mean they actually have that they're the same. It's just there's only so many colors within the color palette to apply. And so you get this smattering of different colors, but each color does represent a distinct population that came out of phenograph. In this case, I ran phenograph, and now we're looking at the overlay of those clusters with distinct populations represented by the different colors. If we look at that same set of phenograph clusters on UMAP, you can see that it gives you a very different representation, right? We've got this group of clusters up here, we got a group of clusters here, and then we got this whole group of clusters over here. And then, of course, TriMAP has another view, right? So each method is just producing an alternative visualization of your high parameter data. How you interpret it within the context of your experimental question and your hypothesis that you're trying to answer with this high parameter experiment is the more important fact, not which representation you choose. Now, we can really look at this grouping of similar phenotypes when we compare, say, T-SNE to UMAP directly, and even TriMAP here, if we gate on just the CD8 populations, and you can do this within your Cluster Explorer module, which is what I think makes one of the reasons why Cluster Explorer is so powerful, because within the line plot, you can actually draw a gate and say, oh, I want to look at my CD8 positive clusters. And so here I've just made a gate around the CD8 positive nodes. Each line represents a different cluster, and you're looking at the higher levels. This is all relative expression on the y-axis. And so each marker, right, has an expression level for each cluster. Each cluster has an expression level for each marker, and we're looking at the means or medians of each marker as these dots. And you can call the phenotype just by looking at one line of color and say, oh, it's high for this and low for that, and high for this and low for that. But all of them are CD8 positive T cells. So if I gate on this line plot to limit what is being viewed in the Cluster Explorer, in the other plots, the heat map and in the two-dimensional T-SNE, UMAP, and trimap spaces here, we can immediately see the problem with T-SNE with respect to how it organizes those events in this low dimensional representation. And as you get into these higher parameter panels, you get splitting of these lineages into these different islands. And they can be very far away from each other on completely different sides of the T-SNE continent, I like to call it. And if you look then at the CD8, same populations within UMAP or trimap space, you can see that they're regionally localized and they're similar to each other in spatial representation here. And so that is why we say that global structure is retained in UMAP, but you can end up with these piled on top of each other in UMAP as well, so you can't clearly distinguish them with a gated region around an island. Well, T-SNE flattens everything, but and within an island you can say, oh, these are probably closely related cells, but you can't infer anything about the relationships across islands or between islands that are far away from each other. Just because an island is far away doesn't mean it's not related in T-SNE. In UMAP, if the clusters are far away from each other, right, then they're probably phenotypically different. Now, going into a little more detail, when you initiate these algorithms with the plugin menu, you'll be presented with a graphical user interface, a GUI here. And with T-SNE had a big problem of being super slow and memory intensive. So it took a lot of memory and it did not process very fast. And it meant that we had to downsample extensively, maybe to just 50,000 or 100,000 events to allow T-SNE to run in a reasonable period of time. And over the years, a number of different groups have improved the T-SNE algorithm by making mathematical changes here. And so I just want to go through these options real quick for you so you kind of get an idea what they are. The first step is that you have to, really the only thing you have to do is choose which parameters you want to be considered during the dimensionality reduction. And honestly, one of the questions was which parameters do you choose? Can you leave out parameters? Can you include ones that you've already gated on? Standardly, I will choose to leave out the live dead parameter, and I actually didn't deselect it here, but you know, if you've already gated for live events, there may not be any additional variance in the negative events that are live. So you might as well drop that because it's only adding additional noise. But all the other markers here are phenotypic markers, and I want to include them. But if you gated down, say you dumped your CD14s, you dumped your B cells, and you're just focusing in on a lineage, well, then in that case, I might not include the lineage markers that we've already gated away because they don't add any discriminating information into the system. So once you've selected your parameters, and I would recommend always using compensated parameters here, of course, you can just go ahead and press run. And the default here in our native T-SNE module is to use the auto opsony algorithm. And this opsony algorithm vastly improves the resolution of these islands when you're looking at multi-million event T-SNEs. And without that opsony option, which actually sets the learning rate, that's how fast you get to a solution, right? And then monitors the comparison of the high dimensional data to the low dimensional data until it reaches a certain fitness and then stops. So you don't even go to a maximum of 1,000 iterations here. It'll actually stop when it reaches a certain goal value. And so it speeds up the algorithm tremendously and gives you better resolution than the traditional Barnes-Hut T-SNE, which you can run just by clicking the manual button here. So I've been showing you all these opsonies because it is now the default, and for most cases, just selecting your parameters and clicking run will let it go. Now, there are options underneath the k-nearest neighbors algorithm and the gradient algorithm. And the k-nearest neighbors is each dot, each event here During each iteration of this algorithm, we'll search for its nearest neighbors. The perplexity is actually the number of nearest neighbors that will be considered for each event. And they push and pull on their nearest neighbors to say, am I similar to you? Am I dissimilar? If they're similar, they'll be pulled together in the next iteration. And if they're dissimilar, they'll be pulled apart. And so as this algorithm cycles through and iterates, this k nearest neighbor finding the nearest neighbors has to be repeated over and over and over again. And originally, the algorithm used an exact methodology called a vantage point tree. And we've come up with an alternative called an Annoy random forest projection, which is a much faster way of finding nearest neighbors. And so if you want the faster run of t-SNE on higher numbers of events, you should choose random forest projection Annoy under k nearest neighbors algorithm instead of the default exact. The gradient algorithm is how much each event moves between each iteration. So the events talk to their nearest neighbors, they push and pull on each other, and they move a little bit. And then it continues and another cycle goes through. So Barnes-Hut was the traditional gradient algorithm. There's an option for FIT-SNE, fast Fourier transform accelerated interpolation-based t-SNE, and that will speed up the gradient algorithm. So the events move in between each iteration a little bit more, and then overall it gets to a solution faster. So if you want to run t-SNE on high numbers of events, you would want to use your op-SNE, which is the default, and then choose k nearest neighbors as an Annoy random forest projection, and choose the gradient algorithm as a FIT-SNE. And that can cut your t-SNE runtime in half compared to a traditional Barnes-Hut t-SNE on the same events with the same number of parameters. So this t-SNE algorithm has been improved to run faster, giving you some options, but it is still very memory intensive. And on a regular laptop computer, I wouldn't try and push more than 1 million or maybe 2 million events into t-SNE. It'll run for hours and hours, but it'll probably give you a solution. If you try to go to 10 or 20 million events, it's overwhelming in terms of the memory consumption to do that type of calculation on a standard laptop. You'll need to go to a bigger computer with more power, more cores, more RAM, memory is the key function here, to run a multi-million event or a 10 or a 20 million event t-SNE. So therefore, you want to downsample to maybe a couple 100,000 events to get t-SNE to complete on your laptop. Now, UMAP, it gives you another dimensionally reduced data space, an alternative to t-SNE, and it uses totally different math, as I mentioned in the comments in response to one of the questions here. And actually, this author, Leland McInnes, actually has a great talk from one of the scientific conferences that's available on YouTube describing the math, and what they do is like build up these simplices, these geometrical shapes, and then create a manifold called a fuzzy simplical complex across your data structure. And I know that doesn't really mean anything to you, it doesn't really mean anything to me, but it is a very different mathematical approach than the embedding used by t-SNE. And bottom line is that it does give you a very meaningful representation of data compared to t-SNE, and it gives you better trajectories or branching of developmental states. It is much faster than the traditional Barnes-Hut t-SNE, maybe comparable to FIT-SNE, but I still think it scales better with a higher number of parameters and the higher number of events. And so you can get away with pushing more events and more parameters into this algorithm and get a result in a quicker time period. And as we've discussed a couple times right now, we get better global structure so that events that are similar to each other will group together in the same region of this space, which you aren't guaranteed with t-SNE. And finally, what we've added recently is the ability to add, to choose the number of parameters you get out of this algorithm. So UMAP, you can create 3 parameters or 4 parameters. And probably the easiest thing is to think maybe you need a z-axis. If you create 3 parameters, then you can look at x versus y, but then you can also look at x versus z and shift and turn the entire structure so you get a different representation of it. And that can help with the layering of clusters being overlapping in this UMAP projection instead of being flattened like you get it in t-SNE. And so I've mentioned that these are different math approaches to dimensionality reduction, but they're still both iterative, cyclical algorithms, and they take some time to calculate because of this nature, the cyclic iterative nature. And so what T-SNE does is it takes the high dimensional data and converts it into a Gaussian probability distribution and across many different sections. And then it seeds the data in this low dimensional embedding. It initializes the algorithm with a certain seeded state. And what the algorithm will do with one iteration is convert this low dimensional embedding, this representation, into a scaled Gaussian probability distribution, and then compare the low dimensional representation, the low dimensional probability, to the high dimensional probability, and ask, are these similar? And they use a math process called relative entropy, or the Kullback-Leibler divergence, to make this comparison. Then each event will find its nearest neighbors, push and pull against them, and move a little bit within this dimensionally reduced space. Then it'll convert to the probability distribution and check again, is it similar? Then it'll move, convert, and check, move, convert, and check. And each one of those is an iteration. So it does this 1,000 times to eventually resolve the positional information for each cell. That gives you your ultimate T-SNE embedding in 2 dimensions. UMAP, it does a similar iterative cyclical process, which is why, again, it takes time for these to run. The high dimensional data is converted into a fuzzy simplical complex. Then the low dimensional embedding is initiated, and how it's initialized is different. Where it puts the events initially is different, and how it compares them is different, but it's still this cyclical nature of the algorithm. So it takes the low dimensional space, converts it into a fuzzy simplical complex, and then compares these 2 complexes into by a method called cross entropy. So again, different math processes, but they still have a very similar iterative process to give you your low dimensional embeddings. And I hope that helps a few of you understand what's going on underneath that progress bar of T-SNE, where it says how many iterations has it taken. Well, it's sorting the cells out, pushing and pulling against each other, and eventually resolving into these islands of similar phenotypic cells. So TriMap is kind of the lone man out here, and it's at the end because it's only been around for a year. It is really a fairly new method, and it's only been a plugin in FlowJo for less than a year. So it does do a great job of giving meaningful representations, and it actually maximizes the variance initially as it initializes by starting with a low dimensional principal component embedding, so PCA. And usually PCA, it's a linear dimensionality reduction algorithm, and our data is by nature non-linear. So we don't usually use PCA for flow data, but it does do a good job of grouping events of similarity, initializing the algorithm. And then TriMap goes and takes selected triplet sets of data and improves that embedding in an iterative fashion, eventually giving you this representation, this low dimensional representation of your data that preserves the global structure. And although I have not really used TriMap, where's my cursor? There it is. Sorry, just going to find my pointer here. I haven't used this on as much flow data as I would really like to judge whether it's as good or better than UMAP as an alternative to T-SNE. I have used it on very high dimensional data in multi-omics experiments where you're looking at hundreds of measured mRNAs and protein measurements together. And I think it's a very powerful tool for showing trajectories or developmental relationships, and it's worth looking into as an alternative to UMAP when you're doing multi-omics experiments. And I'll give you an example here at the end of this deck. Once you have made a choice here and you've created a dimensionally reduced data space, exploring that data space is really the next step. And you can do a lot with just heat mapping across your data, but it's only going to show you one color at a time, one parameter, right? And what you really want to get is that multicolor multi-parameter representation. And so Amlan showed this, and I'm going to reiterate it. When you gate within this data space, this T-SNE space, you can get a nice, powerful way to just scan through the data and look at each of these islands. And you can do that by creating that query gate, creating your overlay with the multi-graph histograms, and then just drag this query gate around into the different regions. Maybe turn on your color axis, focus on a marker, bring that gate to it, right? If you're looking for your rare cells, turn on that color axis, find that little population, and then move your query gate. You can shrink it if you want under your active gate options, make it small, make it big. And then the layout overlay that you've created will automatically update. Wherever you move that query, it'll show the phenotype of those gated cells. Now, the added bonus here is that if you have different conditions, such as this OMIP is a stimulatory, it's looking at a bunch of cytokines downstream of your T cells that are being induced to represent different subsets, and they do this by stimulating with PMA and mycin and compare it to the unstimulated condition. So there's actually two separate samples underlying this being represented here in this T-SNE space. If you gate those apart, knowing what you put into the system as being balanced, 50% from the unstim, 50% from the stim, and if you drop those gates on the root sample downstream of your query gate or any other subset, including cluster populations, you'll get the frequencies of the events where they came from. So in this case, this population, which you can clearly see is a CCR7 expressing CD8 cell with some CD28 on it, something else, some high for CD27. It is predominantly represented in the unstimulated condition. So that means that when I stimulate these cells, they disappear, right? They go away. Almost all of them are gone because they've acquired new phenotypic properties. They've upregulated the cytokines in the panel, and therefore they've moved to a different region. And I know they're still going to be CD8, so it must mean that they're down here somewhere in the stimulated condition. But that gating apart of your samples in conjunction with a query gate can allow you to just mine this data space by moving the query gate around and finding any phenotype of interest based on, focusing on one marker and then getting the combinatorial phenotype. Now, you can subset this data space as much as you want with manual gating. You can take all of these peaks and be like, all right, I'm just going to draw gates and I'm going to by eye subset it out, right? You're going to end up with a whole bunch of populations, essentially multiple query gates, right? And you'll just have to look at them, each one, and determine what's the phenotype and what's the distribution from your experiment or across your experimental conditions there. But why not let the clustering algorithm do it? Am I going to do a better job manually subsetting these out visually by peak finding, or am I going to let an algorithm push out a bunch of populations? So if you let the algorithm do it, then you end up with a whole bunch of distinct populations, and these are represented as these different colored islands. And again, I've overlaid my clusters that come out, which are essentially just gated populations at this point, onto my layout and made an overlay, and then right-clicked and shown the histogram traces, right? Now we're working with higher number of parameters, so more parameters, more complexity. And sometimes it's very clear, a population pops out and you're like, oh, there's my gamma deltas, there's my Tregs, easy enough. Then there's other parameters that have a continuum of distribution, right? Intermediate levels. You can see that with CD27 down here. You can see that with CCR7, right? And the point I want to make here is that with this type of complexity, when you have a continuous smear of a continuous population and not distinct bimodal subsets, manually gating for positivity doesn't necessarily do the complexity justice. We know that these markers are going up and down as part of an immune response. We know that there's going to be transitional states with intermediate levels of expression. And if you just take a hard line gate and chop through, saying everything on the right is positive and everything on the left is negative, well, you just chop through any intermediate expressed population, chop it up maybe into oblivion and never see it. And so again, this is an alternative way. You can manually gate for everything you know in a hierarchical fashion in these data. It gets complex when you have 28 colors, right? But that's still, you know what you're looking for and what the question is you're asking for your experimentals, for your experiment. Making a dimensionality reduction view gives you an overview of all that data, gives you a data space to explore, and then running a clustering algorithm simply subsets in an objective way, not a subjective way, right? When you gate, you're like, oh, I think I'm going to call it here, and that's a line, and whatever's above it is positive. Well, clustering algorithms are going to classify events based on the math underlying them. It doesn't differ from you to the next person, and you just get an overall understanding of the complexity of your data, what types of phenotypes are there, and now your endpoints that you need are what are the statistics underlying those populations, what are the median fluorescence intensities, and what are what's the distribution of each of these clusters across your experiment. So when we get down to asking, well, what kind of clustering algorithm should we use, I'll just give you my personal opinion. And there are a number of good reviews out there where groups have compared subsets of automated clustering approaches. But ultimately, the best ones kind of rise to the top because people start using them, they start publishing with them, and and then FlowJo makes a plugin so that everybody can use them without understanding the coding language where they were originally written. And so we have basically distilled down to three options here. XShift, which is using density estimation and k-nearest neighbors to create these populations within a multi-dimensional space. It finds nodes and then creates clusters based on similar events in that data space. Phenograph uses community detection, and so it's a very different mathematical process. FlowSum is using a self-organizing map, and you can see that even though these clusters aren't one-to-one in terms of the colors, they do cut differently. Like XShift doesn't cut up this group of cells in as many places as Phenograph, and FlowSum the same way. It just has one population, two populations, three populations. And so my general observation that may be worthwhile to you guys is that FlowSum does a great job of finding lineages, but it doesn't do a great job of over subsetting within the lineage. So, you may not get those small, rare populations as distinct clusters out of FlowSum. XShift, it really depends on the data distribution and how much spread you have. And so low spread is better for XShift. Phenograph, it will overcluster. It will subset down to smaller and smaller populations, more so than the other two. And if you're looking for new populations, well, maybe that's what you want. And I'm drawn to Phenograph in both flow data and multi-omics data for the reason that I want to actually get as many populations subsets as possible out of my clustering algorithm so I can find those rare or unique subsets within the data that I may not have known about or gated for manually. So this is just another look at a graphical user interface and going into the XShift. The real awesome thing about XShift is that it doesn't require any other setup. You can just download the plugin and you can run it without setting up R or Python or any other dependencies. And this is Nikolai Samusik's algorithm. It's a Nature Methods paper from a few years ago. This is out of Gary Nolan's lab. And you just pick the parameters and press go is my recommendation, because XShift automatically uses estimates the number of nearest neighbors. So depending on the number of parameters, this number will change. There's different distance metrics, but standardly you just use the default, which is Euclidean distance actually. And then subsampling limit, if you have large data sets, then we can subsample and make it run a little bit faster. But by and large, everything that I show and employ, I just use the defaults and press go. It automatically finds the number of clusters using what's called an elbow method, where it tests a number of different nearest neighbor values, and it finds where the number of clusters where variance across the clusters starts to starts to go down. And so it'll actually tell you, relatively what the complexity is. If you change the nearest neighbors, you can kind of force it to do more or less clusters, but it's fairly stable within about 10 cluster range, no matter what that nearest neighbor K is. So XShift's a valid option. Phenograph is really my favorite these days, even though it's not the fastest algorithm. It can take a while, and depending on the implementation, the 2.4 version actually runs in R. The newer version, 3.0, it actually runs in Python, and it is significantly slower. And so I've been using the version 2.4 Phenograph. I can show anybody or point you to that if you have questions about it. Devil's in the details, but this implementation is running in R, and it does do a relatively good job in terms of speed at producing your downstream population output. You don't have to set anything other than the input parameters and click OK, because it's going to go ahead and run. It uses this nearest neighbor graph approach and then Luvian community detection to partition the graph into those distinct populations. And as I said before, Phenograph seems to over cluster, making smaller and smaller populations. Intermediate levels of expression can be subset into distinct populations. And so out of all these methods, Phenograph seems to subset even deeper than the other two. And so if you want a lot of clusters to work with, in this case 39, you don't have to set that number. You just press go, and Pheноgraph's going to return as many communities as it finds in your system. And then again, your job is to phenotype those community populations and understand how they change across your experiment and your experimental system. FlowSum has become extremely popular for the main reason is that it's the fastest by far. It is super, super fast, and you can classify millions of events without any issues. I mean, it might take 30 seconds or a minute when you put 2 million events in here. And while other algorithms will be working for hours, FlowSum, it's so fast, this self-organizing map approach, that you can really classify a lot of events in a reasonable period of time and move on with the enumeration of their phenotypes. And additionally, FlowSum comes with, it outputs some visual representations of the clustering, including a minimal spanning tree with radial star plots that allow you to get an idea of the phenotype within each of these mini clusters. You actually start, it's got a whole bunch of options that have been unlocked here. It starts by creating a self-organizing map in a grid, in a 10 by 10 grid. So each one of these little circles represents one of the mini clusters, one of the 100 mini clusters that gets produced. And you can change that grid to be larger if you have larger data sets with more complexity. But you have to actually specify the number of clusters. And I say you have to because if you use the auto option right here, it'll find a few clusters, but it's typically a very low number. It does not do a good job of over subsetting or over clustering. So using the auto function is not, you aren't going to get 39 populations out of here. The only way you're going to get that is by specifying the number of populations as the number of meta clusters. And so these 100 mini clusters get grouped into meta clusters by their similarity, and that's what the population is. So each of the colors, the groups of colored nodes, represents a distinct population that's coming out of FlowSum here. You get a heat map if you as an option with dendrogram and sorting the populations based on similarity, which can be helpful in interpretation then. And these are both images that come out of R. When you run FlowSum, it sends the data to R, runs the R package for FlowSum, and returns back all of these cluster populations and the visuals to you. So you can lay out this set of mini clusters and the overall meta clustering view either as a minimal spanning tree or as a grid showing the 100 in a square grid. And that's actually what EmbedSum is doing. It's laying out the data in that grid form of the FlowSum clustering output. So don't be afraid to try each of these. FlowSum will be your go-to if you like have an idea of the complexity of your data and you know how many populations you want to get out of it. Then you can just put that number in, press OK, and go and get those populations. If you want to over cluster, I would say again, go to Phenograph and let it run and see how many populations does it produce and how does it cut up some of those lineages that FlowSum may not have cut up, and are those distinct populations. XShift, always worthwhile. It's kind of not so, it's not slow, it's not fast, and it does a good job of subsetting. I guess the last thing I'll say about FlowSum is that you can actually take this map that you create, this FlowSum self-organizing map. When you run it the first time, you can actually go back and use that map again by choosing the map under Apply on Map. There'll be a drop-down menu here. When you select that, you'll see the last run of your FlowSum. If you select that previous map, the option for clusters will gray out, and the only thing you can do then is click OK, because you're using the same settings and you're actually using that map to classify new events into the same FlowSum classifications, into the same clusters. And so unlike these other two algorithms, which you have to have all the data present to cluster together to make it comparable for Phenograph or XShift, FlowSum, the way it's coded, you can actually apply that map, that new data, to that same map, or classify different data. So you could go back and say, look, create FlowSum clusters on your concatenated file, and then go to all your original files and run FlowSum on each of those original files, classifying all the events. Even though you left some behind because you downsampled, you could classify all the events in each file, and then you get different views or minimal spanning trees and relative levels of populations here, and I'll try to give you an example of that. No matter which method you use, you can spin up Cluster Explorer, and actually we've done a new release of Cluster Explorer recently, so it's been modularized into distinct windows, as Amlan showed you in his demo. It doesn't look like a single panel the way we had it here a while back. And so now you can close and open these windows if you need to. You can make as many 2D plots as you want. You can actually compare these different dimensionality reduction methods and choose different cluster sets from your control panel here. And I'll show you that in just a minute. So I want to take this a little bit further because, yeah, sure, we can find these populations, and I'll show you how to get the expression across your different conditions. But what happens when you found a population of interest, right? With a clustering algorithm, you don't have any hierarchical gating to get to this population, but you may want to sort it and purify it for some downstream application, functional testing, or going into a single cell RNA sequencing experiment. And so the question becomes, how do you sort a population identified with a clustering algorithm? And the method that we've come up with is to employ this approach called Hyperfinder. And this is actually, Hyperfinder is our plugin employing two different methods for creating gates to find a population. Two publications that came out about 2 years ago. One was called Gate Finder and the other one Hypergate. One uses convex polygons like I'm showing here, another uses hyper rectangles in the data space as a starting point. But what Hyperfinder does is it takes the data and it splits it up into a training and validation data set. So we actually first train the algorithm to find the events in the cluster by building this biaxial gating sequence across all potential dimensions. And then it goes through and it checks, well, are my data in the cluster inside the gate? And it picks a sequence of gates that it will find that maximizes the overlap, which is how many events are found within the final gate compared to the training data set. And then it goes and starts moving the gates and optimizing the boundaries. So the blue is the optimizes what's being shifted with each iteration. The gray is the original gate, and blue is being optimized. It's shrinking the gate and moving its position, changing the vertices, trying to, and each round as it optimizes, it goes through and it measures calculates the precision and the recall and the F measure score, which is the harmonic mean of precision and recall. It's like a measurement if you gated and I gated, what is the overlap between our two gated populations? An F measure of 0.9 would mean 90% of the events that you gated in your final population are also in my final population. So it's a measure to look at how much overlap there is. And so what this is doing is trying to maximize the F measure score over time by iterating through and changing the boundaries. It then like cuts out any bad gates that don't improve the overall F measure, and eventually comes up with this hierarchy of gates, which it creates in FlowJo as gated as actual gates on the sample. And it gives you the recall, the precision, and the F score, saying, well, what's the final result of that? How many events can I capture with this gating hierarchy? So it's building a hierarchy for you without any previous knowledge, using totally mathematical approach and trying to maximize the overlap or the capture of those events. And it actually goes back and validates using another subset, and that's what the recall, precision, and F measure are given back on the validation data set, not the data it trained on. And so what this means is that you can go through and you can say, oh, maybe I can only sort on a subset of parameters using my sorter. And so what I'm going to do is run Hyperfinder to find this hierarchical gating sequence that ultimately gives me a high purity, a high measurement of of the events that we get back in. And in this case, this is actually an intracellular cytokine staining assay, but just using the surface markers, if I want to purify that population, well, I can do it in 4 gates based on Hyperfinder, up to over 95% purity. That's what this measurement is, and it only took 34 seconds to run. So this is the output that it gives you at the end with the precision, recall, and F measure score, and how many seconds it actually took to find this gate sequence to purify that population in a hierarchical manner. And so what does this mean? Well, now we need to actually sort this population, right? So how do we get those gates out of FlowJo and into our sorting software? Well, we've added an export to fax diva function within FlowJo, so you can actually export your gates to a diva experiment and then go and sort that population. There is some details there if you want to be walked through that. Of course, I can help you with that, or one of the other technical application specialists. But bottom line is that we can actually export our gates into a diva experiment and then sort. So you can imagine, and this is completely a real world use case, might take 10-15 minutes on 100,000 cells to do the whole process, is to run a clustering algorithm, right? Run a dimensionality reduction algorithm and find a population. And then what we do usually before running Hyperfinder is kind of clean up that population so you get the core population, not the outlier events. Run Hyperfinder to find a biaxial gating sequence, and then export those gates back to Diva so we can sort that population. And then you can work with that population downstream. You can also use Hyperfinder to look for alternative approaches to gating that population, maybe making more pure, maybe dropping a few parameters in your sort, because we always come in to our hierarchical gating with some preconceived notions that we do this first and that first and that first, and Hyperfinder doesn't enter with those preconceptions. It just wants to find the shortest way to get the purest population possible. So now that we have this population, right, what are we going to do with it? It could be a functional study, it could be growing those cells, cloning them, whatever you need to do downstream. But you could also take that into another experiment using deeper multi-omics technology and looking at hundreds or even thousands of parameters at the single cell level, employing single cell RNA sequencing. And so if you don't know about this yet, it is a hot topic in immunology, and many groups are rapidly developing their ability to use this technology so that they can actually look at the RNA transcription profile of the cells that they sort. And it's an expensive technology, so that's why we sort, because the most expensive piece is actually the sequencing. And if you sequence hundreds of thousands of cells, well, that's going to cost a lot of money. And so what they usually do is sort the populations, and then we can tag them with fluorescent antibodies first to sort, right? And also sequence tagged antibodies so that we can actually sequence the protein readouts. And we even have a multiplexing kit, so you can put together 2 or 4 or up to 12 different samples all on one chip and sequence all that together. And so the workflow here is to, find a gating hierarchy that sorts the population of interest, do your staining of your cells, then sort using the fluorescent tagged antibodies. You take those sorted populations, and in S6 you can do 30 parameters up to 6-way sorting, right? So you get 6 different populations. You can drop all of those onto a little chip. This is the Rhapsody system. And the chip is a microwell, and the cell goes down, and then a bead goes down, and you lyse the cells, and all the RNA as well as any sequence tagged antibodies get captured by the bead. You do your library preps and then sequence that. And we now have a SeqGeek application, which is very much like FlowJo for sequencing, that allows you to do dimensionality reduction, clustering, and differential expression analysis to get at the subsets of RNAs or proteins measurements that we've probed here with this technology, and understand what the combinatorial expression is for distinct states within your cell's subsets. And so just to look at SeqGeek really quickly, it looks like FlowJo, right? With a green tint to it. Instead of groups, we have gene sets, which are collections of genes that you can make manually, or you can create using what's called a volcano plot. And with a volcano plot, you're essentially comparing 2 populations of interest for genes. Each dot is a measured, 1 or more measured features here, not cells, in the volcano, and all of the features that are very high here and up in one population versus another, or up in another population, down in the population versus another. When you gate within this space, you create a gene set which actually gives you these features that are differentially expressed. And so we can demultiplex our populations, and we can do dimensionality reduction and clustering on top of them. Just as a detail here, we have 2 views in SeqGeek. One is a normal view that you gate within, or you can see the distributions of events across your measured parameters. That can be RNA and protein measurements here with AbSeq. And then we have a tab up at the top of our graph window in SeqGeek where you can shift to a gene view, and that essentially pivots the graph over, and it takes each of these individual cells grouped as a population and sums the expression for each gene, so you actually get an overall expression level of the population for each gene. And so what we're looking at here is a balancing act between 2 different populations. Each dot is 1 or more measured features. Those features that are on the diagonal are equally expressed between the 2 populations, and those that are off the diagonal are differentially expressed. And you can gate within this space, or you can actually click this extra button up and down arrow to get to your volcano plot. The volcano plot actually derives the fold change and the p-value, or its q-value, which is a modified p-value. It's modified for the number of tests for false discovery rate. So it's a higher threshold than the standard p. And what you're getting here again is the ability to gate on the features that are up or down in those populations relative to each other, getting a gene set with all the statistics of how much, what's the fold change, what's the Q value and such. So if you double click on a gene set that you've created, you'll get all those statistics. What's the parameter, what's the fold change between your different populations, and what's the statistical significance. And this is essentially one of the endpoints, because then that gives you a collection of genes that is the feature set of that particular population. And I noticed somebody ask in our Q&A and our list there in the chat, can you use these for multi-omics or single cell RNA sequencing? By all means, you can. And you actually have to, because once you get to working with 400, 500, or even 20,000 parameters, if you do a whole transcriptome assay, well, that's way too many parameters to even consider doing sequential gating on, right? And RNA isn't expressed at the same level as protein, and so you can't lineage gate on RNA in the same way you can on protein. So if you have AbSeq in there, if you have protein measurements, you can still do some gross hierarchical gating on single-cell RNA data and multi-omics data, but most of the time people use dimensionality reduction as a first step. I'm showing a sequential dimensionality reduction because T/E won't work if you throw 400 or 500 parameters at it at the same time. But you can do sequential where you run principal component analysis and go from hundreds or thousands of parameters down to 15 or 20 principal components. And then you do T/E, and the input of this T/E is the principal component parameters, not the raw parameters, the raw measured features. So you T/E the principal components, and that gives you this nice visualization. So these are the different subsets that we sorted on the S6. You can see they're nice and cleanly separated. Some of them are NK cells, some are B cells, some are T cells. And I'm doing this with all measured parameters. In this experiment, we had about 30 protein markers and 400 RNAs. It's a mouse-targeted immune panel. And what I wanted to demonstrate here with this image is that you don't have to just look at everything. It's similar to gating down to a lineage and then focusing on it, as we've discussed earlier tonight, or this morning for you guys. But now you can take any of those subsets. You can merge them together using Boolean gating, or however, I've got these subsets and I can merge them together and look at all of my NK cells separately, right? I don't have to always look at the wide angle lens view. Instead, I can zoom in by focusing on a gated set of populations and then using an alternative method to spread them out within this dimensionally reduced data space. In this case, I'm using TriMap, and as I mentioned, TriMap is very successful at giving these kind of long trajectories, and the way these cells are connected to each other ends up being visualized in these lines connecting between the major populations. And so if you go and take that TriMap space, right, with all your NK cells and run phenograph on it, well, you get distinct subsets, distinct populations with distinct transcriptional states. And there are various plugins and methods that we can employ within SeqGeek to do differential expression and ask, well, what are the genes that are differentially expressed in cluster 6 compared to all of the others, right? And what you get are gene sets, and those are the targets that are enriched in those populations, right? POP6 is up for IRF8, right? It's a functional marker. All of these CD11B high cells are high for LGALs. CD7 is high in the middle. And these are just 3 markers, but you can start pulling apart whole combinatorial phenotypes at the RNA and protein level together and get a signature for your cluster populations, which then you could go back and verify via flow cytometry if there's any new targets in there. Maybe you can do pathway analysis or look for overlap on of published gene sets to understand what are the biological functions of those genes and which pathways they fall into, and infer functional information about these populations of cells from the expression repertoire that you get out of these types of high dimensional panels. So that's all I have in terms of my presentation. I'm probably already well over time. Everybody's going, oh my God, Tim, finish up because we all have to go to lunch or something. But in summary, I just think that we have started to employ these plugins as a mechanism for bench scientists without bioinformatics programming experience to use algorithmic tools. And we essentially allow you to point and click at data, call out to an algorithm, run the algorithm in that environment, R or Python, and return back those results into FlowJo so you can interact with those results again. And we've presented a workflow which enables really a mining of data in a objective way, and enables discovery and potentially even isolation using Hyperfinder and export to Diva of those complex novel populations in your data. Linking together our acquisition software with our analysis software so that we can transfer gating allows us to actually sort those cluster populations using computationally derived gates. And if you haven't seen it yet, the single cell multi-omics data is very impressive. We have our system called the BD Rhapsody, which is a microwell system. It allows you to do both protein and RNA measurements in the same experiment. And then we have an application called SeatGeek, which gives you a FlowJo-like point-and-click interface for analyzing the results of those experiments, providing this full solution for single-cell multi-omics analysis. Whole bunch of people. Of course, I'm not going to go through all the names, but thank you to all the data and algorithm authors and my apps team who actually produces data for us to use in these presentations. And of course, my informatics team that codes these plugins and allows us to use them without knowing the programming languages. So with that, I'm just going to hop out. I think we're actually out of time, of course, but I do want to hop over for 1 second and take a look at this experiment and just show you kind of the way I go about this. In this experiment, I had 16 original samples, and I started in a different workspace. And what I did was run FlowAI on the root samples in another workspace, and then I exported only the good events coming out of FlowAI. So I don't have an actual FlowAI gate here, but I use FlowAI separately, and then I just export the good events and bring them in as new files to work downstream. So I'm essentially using a workspace to clean up the data initially with FlowAI, and then working downstream with those events. Within my FlowAI good events, I can gate down to specific populations. And maybe I want to clean up singlets, get rid of the leftover unlicensed RBCs with a CD71, exclusion gate. And then in this case, I'm focusing in on all the CD45 positive cells. And so what I do is actually take each of those samples and make keywords that describe where they are from in terms of which time point. You can see the dates here are different for each 4 samples, time point 1, 2, 3, 4. It's a time course of this animal across and probing different tissues across that 8-day time course. And so we've got lymph node, liver, spleen, and blood samples. And this is kind of the example that you've seen in the presentations. Well, if we take all of those samples gated down to good CD45 positive events and merge them into our concatenated file, we get this file, right? And this file, you can see I've already run a number of different dimensionality reduction algorithms and clustering algorithms on the same root file. So I've got flow sum clusters, phenograph clusters, X-shift clusters, a trimap, an embed sum, and a UMAP. Maybe we can see them all right here. And compare DR methods, right? So different methods to visualize that data downstream. And the grid that I showed you earlier, this time point versus tissue, these all come from these derived parameters, I'm sorry, these keyword-based parameters in the original cleaned files. I've given 1s, 2s, 3s, and 4s, 1s, 2s, 3s, and 4s, and that gives positional information for those events. And so the sample that has a 1 and 1, well, it's right here, right? Those are the events from that particular sample that was a lymph node. 2 and 1, right? 2 and 1, that's going to be my second time point, or sorry, second tissue there, 2 and 1. And so that's from the liver, right? And you can go across and you can organize your samples into meaningful representations. And then when we gate, we're going to gate apart the different tissues or the different time points so that we can compare those frequencies. Now, usually I'll go ahead and run dimensionality reduction and then derive some populations. And what I always used to do is take those populations off of the concatenated file. So here's all my flow sum populations down here. And you can take them and highlight them and export them as individual samples and bring them back in to FlowJo. Just like any other sample, you can add a bunch of statistics, in this case the counts, the medians, and then I'm dropping the gates for my different conditions downstream of them. And when I create a table of this set, right, these are my flow sum pops, create a table, well, that's all the statistics that we need in the order that we want them to be in. So these are flow sum population. I'm focusing on phenograph here, sorry. Phenograph populations, and then in this table, 1 through 32 here, and then these are all the statistics, the counts and the median fluorescence intensities. And at the very end, we have the frequencies across the different conditions in our system. And so that can be formatted in Excel just by copying it out into a heat map. But instead of going through that process, now we have the Cluster Explorer. And so instead of doing that, I would just go to my concatenated file and spin up Cluster Explorer, Workspace tab, Plugins menu, right? Here's my Cluster Explorer. And here I'm going to leave all 3 cluster sets selected. Maybe just deselect one marker that I already gated on. It's the live dead, I think, in there in blue, or in the BUV. And then keep all of these parameters because I'll get multiple accessory plots. Cluster Explorer will gather the data, and it'll present you with this multi-window view where you can now interact with the data. Cluster size plot, line plot, which again you can select any subset. So where are my NKG 2D positive events? Well, once you gate or select any subset, gate on a subset of clusters, it'll highlight only those clusters, or it'll limit the highlighting and visualization to each of those clusters, and you can see where those actually live within your 2-dimensional data space. And so let's get to there. Just look at those two. So here's my T-SNE, and here's my UMAP, right? And I can even move this around and drag the gate. There's so many things you can do with Cluster Explorer. Take a look at the control panel up here, and you can see there you can choose different cluster sets. So I'm going to double click and reset this, then choose a different cluster set. I'll look at my flow sum clusters, and everything changes, right? The MFIs change, the visual overlays change, and the heat map changes because we're looking at fundamental different set of events, or a different set of clusters on the same set of events. Go to my X-shift clusters, right? They change as well. Go back to my phenograph clusters. And you can look at each individual cluster by clicking on it within the dimensionally reduced data space. You can multi-select these by holding down the control button if you want. Again, you can gate under the profile plot gates. You can choose and or not. So maybe I want to take a look at only CD19 positive B cells, and then that are not CD127, right? That limits what I'm seeing, and are not CD95. It removes those clusters, and now I can see just what they are. Where's that CD95 positive population? Let's delete that gate. There it is, right there. Right? Click on it. You can change its color. But you can use these gates to limit what you're viewing, and then the heat map shows you the same thing. I like to choose medians and scale values. You can actually just, here, I'll double click it and clear the gates and play through the clusters with the button, right? Just seeing what's in my system and comparing the visual representations of the different dimensionality reduced spaces. But what's also nice is that once you have a visual representation that you want to look at here, you can always right click and copy that image. You can copy any of these images. You have a bunch of options to change. You can look at the expression level as heat map, or you can look at global density, which is a pseudo-color plot. But all of these images can be copied and pasted right into a presentation. Here in the heat map, you can actually make it larger to see your column headers. And as Amlan showed, if you want the distribution of that cluster across your experiment, well, you need to pick the gates. Add nodes as columns will essentially drop those gates downstream of each cluster population and give you the frequencies across your gates. So in this case, I have my blood, liver, lymph node, and spleen. And I'm going to go ahead and select each of those, right? Select those 4 conditions. Remember, I started with 25% of the events coming from each of those conditions. And so when I select those, each of these values underlying this heat map across the blue to yellow, blue being high, yellow being high, blue being low, and green being intermediate. This is a pure scale of 0 to 100 here, so it's just frequency. A mouse over the cell will give you the information within that column, the number of events and the frequency. Well, this cluster 1, 72% of these events come from the liver. So you can know that this particular subset, which is a CD56 positive CD8 cell, so more on the innate side, comes from the liver where I would expect it to be. Well, cluster number 3, 93% of those events come from the blood, right? So there's the phenotype and there's the distribution. All of these numbers, the underlying data, can be copied as content and pasted into Excel for downstream use. You can also copy the image here. But essentially, you get the phenotypic property of the cluster, and then by adding these additional nodes as columns, you get the cluster's distribution across whatever experimental condition you decided to gate apart. Whatever you put together in your concatenated file, you can pull it back apart and understand its distribution. So I think that's a much more direct route, just spinning up Cluster Explorer and going to the heat map and exporting your numbers, as opposed to exporting the clusters and then actually dropping all the statistics on there and enumerating them in our table editor. And that's where we're going. Now, ultimately, I may want to sort that population, and so I want to focus on that cluster. I would just go to the concatenated file, find the cluster population of interest, go back to my plugins menu, and run Hyperfinder on that, right? Oh, I'd have to do it on the root, sorry. So root, Hyperfinder, and then you pick the actual cluster population within the UI of Hyperfinder of which one you want to do after selecting the parameters. So I'm way over time, and I think that we could do a separate session on just any of these different subsets of topics. But I'm going to return the ball back to Amlan so that he can wrap up with you guys and get out whatever information he needs to, so that you have some data in hand. And what they're going to give you is a 25 color experiment, right? That has all the major immune populations. It's these 25 color FCS files. There's 5 samples. And what we're going to ask you to do is follow along the set of instructions where you bring your data in. You actually are probably going to need to edit the columns and show those additional keyword columns that we have added. So patient ID and experimental condition. Just as an example, I've already exported with these keywords in there. And as Amlan showed, we're going to have you select equivalent nodes, go ahead and export the populations as a concatenated file using a small number of events, and create those additional keyword parameters so that we can pull apart the individual samples. And this is just an example experiment, right? But it is the same workflow whether you're using your own data or this particular example data set. We're going to create that concatenated file, bring it back into our workspace, and then we're going to use that file downstream. Here's our concatenated file, and we can do dimensionality reduction and clustering on it as an example, so you get a feel for the graphical user interfaces. If you want to use their R-based tools, all the instructions are to use just XShift and tsine, which do not require R. But if you want to use FlowSum, Phenograph, or UMAP, or actually not even UMAP, but FlowSum or Phenograph, you're going to have to download and install R, which I believe the applications team is going to assist with as needed. So I will stop talking because I could talk all day, all night, and forever. And I'm going to say thank you guys for your time, all right? And we look forward to seeing you again next week. Go ahead, Amlan. Take it back from me.
Yes, thank you. That was fantastic session, Tim, and some really cool stuff was about to happen, but just because of the time crunch, we couldn't do it. So I would request in next session we can have a small, like half an hour session where you show us some of these stuff like Hyperfinder, what you're about to show. And then I've discussed with Keith also. Keith is okay with that. He can take a, I mean, he can continue with his part after you finish. Is that interesting? Sounds? I think you're on mute.
Sounds like the plan. I can show a few more things on the next session before Keith goes. Thank you.
Great, great. That's all from my side. Peter, you want to put some closing comments?
Sure, sure. Thanks, Amlan. That was a fantastic session. And Tim, I know you can go on. We are all excited to continue, but then people have plans, and that's why I've seen some 10 people dropping out. So I'm sure we look forward for your session in the next time, in the next plan when we have. So before we close for the day, a small announcement which Dr. Sumanto would like to do on the breakout session. Sumanto?
Yeah, thank you, Peter. So, participant, we expect that we will help you in installing the R package and checking the functionality of your plugins. So we are having a breakout session arranged on Monday, 8th of February. So time will be between 10 to 12, and there will be another slot in case the first slot is not you are unable to join in the first slot, or still there is any difficulty, you can join in the second slot, which is in the same day, 2 p.m. to 4 p.m. So the idea is, you try at your end to install the plugins and see if it is working, and try to install R. Still, we'll be having the session for you on Monday, and you will get a joining link for that. Ankita, will it be a separate joining, or Ankita
Sir,
can?
it will be separate joining both the sessions, as we have mentioned that there are two sessions on Monday itself. So there will be two separate links. It's up to the participants, whichever they want to join. So it's up to them.
Right. So it will be just to see the installation, the step-by-step process to install the R package and the plugins. And still, we will request you to try at your end. Sorry, try at your end the procedures and all you will find in the FlowJo Exchange site itself. And if you find difficulty, please join in this breakout session.
Yeah.
Thank.
And thank you, Dr. Samantha. And before that, please get your FlowJo software connected, downloaded, and functional, and then you can have your plugins also installed into it. I'm sure the invites have been sent to most of you all for the access of the FlowJo, or you might be using your own FlowJo. I'm not very sure because I have not seen all 100% acceptance of the links. So probably you must be using your own FlowJo. So for the plugins, before you get into the plugin session, ensure that your FlowJo is functional. You have a function FlowJo on the same computer which you are using, and then you can join with the plugin session. So with that, thank you very much. Thanks,
Peter,
Tim, once
sir,
again. Yeah,
just one
yeah.
thing. Would you like to tell them that what from which email ID they will be receiving? Because few are mentioning that they haven't received, maybe they have missed out. So it will be great if you could tell the email ID from which they are getting the
It's coming
FlowJo
from the
access.
FlowJo. They're getting it from the FlowJo because those people who have not received it, they wrote to me and I've sent it to them and they have got the access and they have conveyed back also. So if you have not received it, you can write back to, I think they've been writing to BD Biosciences, right, Ankita, right?
Right, sir, right.
Or
Yeah.
they
Yes.
can write to me. You can share my email ID, Ankita, on chat. Okay,
Sure, sir.
Ubaldo underscore Barbosa at
Yes.
bd.com. And then, okay, so the email IDs which we are giving access to is only yahoo.com, yahoo.co.in, and gmail.com. Only these three email IDs will get the invite or the links. The other email IDs with the institute ID extensions are not getting the this, okay? Okay, so with that, thank you very much. Thank you so much.
Thank you.